ways that like I think normal people aren't, and they're really the worst of society. And it's really the reason why I think the United States is spiraling downwards right now is because of people like this.
Damn. Okay. That's interesting. Yeah, well, I mean, I can definitely see from someone like your perspective why you'd feel so strongly about that. But anyway, yeah, I gotta get back to my workouts. But I think at this point I was listening to another YouTube stream, but
Hmm.
I might just listen. This might actually be the best content right now live as we speak. So I'm gonna tune
There
into
you
this
go.
while we work out.
Thanks, dude.
Yeah,
I'm also streaming on YouTube, so you can like just pop on over there too, and you can pause and stuff too.
nice, nice. Sounds good. All right, Dr. Greg, have fun with this one.
All right, thanks, man. Appreciate you. Peace.
Yeah.
All right. Hi, YouTube. Dr. Greg Show, December 8th, 2025. Welcome in, guys. We were introduced to a terrain theory channel yesterday, and oh man, guys, it's not good. It's not good. It blows my mind how successful these people are. How, thankfully, I don't know. Maybe he bought some viewers, subscribers on YouTube, because his other stuff is so small. But then again, I have a really small audience on anywhere other than TikTok. So like I know that this has happened. It's kind of hard for people to follow your content necessarily. So, but yeah, it's shocking. It's shocking. We got a person on the left that was a self-admitted biochem dropout, went back to school for psychology, and now he doesn't believe in the entire field of microbiology. And then and then you have on the right, dude is an journalist that doesn't believe in viruses, but he believes that SARS-CoV-2 is real, probably because he thinks that the Jews made it. And he's now going to be trying to convince this guy that the virus is real, even though vaccines aren't real. Because it's so funny. Yeah, it was recommended by someone, and I made the connection early on. One of his early answers was very like weird, and I was like, whoa, it's like this is rhetoric. This is anti-vaccine like rhetoric. And then sure enough, I go into his stuff, and I mean, it's right here. You know, I'm an independent voice. I'm an independent guy. Independent, independent, independent. Just like this is the one that we go on. This is his website. This is the thing that he must have gone after, and like that was the thing that he must have like said or something in order to get the ire of this guy, I guess. I don't know. So there you go. And here is his independent state. Here you go. Scientific data show that unvaxxed children are healthier. Yada yada yada. I'm an independent researcher, journalist. And then he just has all these anti-vax stuff in this ACIP meeting this last week. He's just been retweeting all that junk. He's a huge fan of Aaron Siri, of course, because all these people are, you know. Yeah, Bill. I mean, look, I think if you have cautious optimism or cautious skepticism of, media organizations, that's fine. Like, that's cool. But extend your criticism towards places like this guy who legitimately has no idea what he's talking about. And I feel like these independent voices get off, easily, because nobody actually critically engages with them. They're able to kind of like withstand and be in this morally ambiguous area where they're like, oh yeah, I'm just looking out for the little guy, like the big pharma company big pharma company bad, and everyone's like, oh yeah, that's a good point, yeah. And it's like, That's not really what guys, we're not pro-vaccine as much as we are anti-virus, anti-disease, right? I mean, if there was some better way of doing it, I would be for that. I don't care about the pharmaceutical company making it necessarily. Like, I don't care about that. I don't care about protecting their profits. Like, I think that we should have nationalized vaccine production, and we should have patent-free vaccines so that pharmaceutical companies can't. Like, imagine that. Like, why? Yeah, like, holy shit, guys! Like, it's insane to me that we let these guys, like, aluminum was not gone from our vaccines. You know, you're talking about aluminum being a neurotoxin. I mean, it's just thing after thing. Aaron Siri again, a big thing about like, like it's with the fedora hat, guys. With the fedora hat. You ever gonna trust this guy with anything? You gonna trust this guy? Is this who you're gonna trust with your health? Huh? This guy right here? Think about that. This guy's looks like a psycho. Okay, sorry. It's so easy. It's so, guys, like the path forward is so easy. To be a huckster, to be a grifter, to be and all that kind of stuff. It's so easy, and it's ridiculous. What's up, Amby? How's it going?
I just want to sit in that for a second. Like, that guy genuinely, because I laughed out loud when you said that, because I was thinking the same thing. And sorry for my background noise. I'll hop down in a second.
Okay.
But that guy genuinely looks like a psycho.
Dude, you're amazing, Bear. Guys, we're up to 23. 3, 2, 1. Guys, we're up to 1,023 political actions now. You guys, we have done it. We are past 1,000 on both of our trackers now. How insane is that? It's amazing. So cool, man. It's so cool. Thank you, guys. Helen, you guys are both legends. Absolute legends.
Let's go. It's amazing. See, and that's what we need to get guys like that away from our healthcare. Because look at that guy. That guy probably doesn't even know what an atom is.
Like, I legitimately, I don't think. I mean, we gotta see. We gotta see what he says, what he does, right? It's up to him to make me look like an idiot, and maybe he's super well versed and well researched. Looking at his Substacks, it's not. You know how my point is that it's okay if people don't have the degrees, and I'm not even trying to gatekeep. Like, legitimately, I don't. That's not the point here. You know, you could be theoretically a journalist that is sourcing legitimate scientists and looking at the research and going to that, and you know, like, you know, that's awesome. But they're not doing that. They're going against about it. Yeah, exactly, Mackenzie. It's like it's the opposite. They're sourcing just trust me, bro, and they're trying to take down all of medical science. So
just to touch on that really quickly too, the reason that they just trust me, bro, is because they distrust the medical science so much. So all that they believe is the other people that have either similar lived experiences to them or whatever. So people are saying 5% of the truth, like we've talked about before.
Sorry. Damn it. Sorry. I'm sorry. Your last sentence one more time, please. I'm sorry. I didn't mean for that to play.
So then, you know, these grifters, even RFK, but just talking about these grifters for a second, and then I'll hop down. They'll say 5% of the truth, and then people resonate with that 5% of the truth, and then the rest is just BS.
Exactly.
So that's how they get them. Yep.
And look at this, guys. He actually interviewed this researcher, independent journalist, beacon of truth and power in the world, is actually he interviewed RFK too. So thank you for MB. Thank you for coming up. I'm not American. Who's that guy? Because he just Googled his name and he's a hacker. I don't know. He's an independent researcher, journalist guy who believes that SARS-CoV-2 was real, but he's anti-vaccine, and he's going to teach this guy that terrain theory is not real. Like that viruses are real. Guys, I'm gonna like start going after these guys a lot more, like in my stuff. I've made the decision that I've learned how to stitch and duet, and I have the technology now. I figured it out. Okay, I'm so sick of these guys, and I am actually really mad, and I legitimately am furious about it, and I'm gonna absolutely be coming after these guys. So please, thank you for everything. Yeah, thank you, Mackenzie. We gotta watch the video, but okay, what's up? Hi, Stealth Cats, how's it going? Hi, Miss Cake, how's it going?
Hi, good. I just received my copy of Science Under Siege. How to Fight the 5 Most Powerful Forces That Threaten Our World.
Peter Day.
Just reading the foreword now, but it's already really excellent.
Nice.
I recommend it.
Hotez, right? Hotez and...
Yes, Hotez and Man.
Okay, nice. Yeah, it's really... Hotez is legit. He's one of those legends. He developed an off-patent SARS-CoV-2 vaccine. Like, he did it. Like, he's done it. He has an MD, PhD. He's incredible.
So, enjoying it so far.
Nice. Science books. Yeah, Hotez is awesome. I totally agree. Thank you for coming up and saying that. I appreciate you. All right, well, it's an hour and a half, guys, and I know, isn't that amazing? Look at that, shaboom! I love it. All right, guys, here we go. 3, 2, 1. Here we buckle in. Yeah, buckle. I was gonna say buckle up. Mackenzie, Jinx. You got Jinx.
Welcome, everybody, to another episode of the Beyond Train podcast. I'm your host, Liam Dalton. Today, we have a very special guest on. I'm very much looking forward to this discussion. We have Jeremy Hammond on, and we're going to talk about viruses today. I'm really looking forward to this discussion. It's been a long time coming. To be quite honest, it's been kind of difficult to get somebody on to talk about this from the opposite perspective. So I'm really, really grateful for Jeremy.
Let's see how easy it is for me to get on after he says that. That might be a age like milk scenario.
In his time today.
Stealth, yeah, it is.
And if anything, I think in a true pursuit of truth and in real science, it's best to understand both sides of the argument. It doesn't matter where you fall in your opinion. It's best to know both sides to be able to not only strengthen your argument, but gives you a true holistic understanding of what's going on. And so I think,
dude, true science, holistic, like he's hitting all the buzzwords. Even in his own explanation of science, he has to use his terrain theory mumbo jumbo in order to say it. He's just unbelievable. Yeah, exactly, Hazel. Facts on both sides. Like, what does that even mean? God.
I think that's really important. So
see, you guys, I'm like, this is only a minute in. I got to 5 minutes, and I was like, I have to stop it. I can't do this any longer. I can't do it.
If everyone keep open mind today and be receptive to this, I think that's one of the most beneficial.
Keep an open mind.
Things that we can do moving forward. I don't care if you're no virus or not. I think that's really important. So, anyways, I've talked enough. Jeremy, thank you so much for coming on today.
Sure. Thanks for having me.
Definitely. So the question I ask all my guests, the introductory question is, what is health? And sort of get a definition of what health is, what it means to you, how does it manifest? And this gives us a good little baseline to work off of.
Gosh, what is health? There's so many ways you could describe it and define it. I guess basically I would say that a state of health is a state in which your body is functioning as it was designed to do.
Here it is.
I guess that would be my most simple definition.
Watch this.
Nice and concise. Perfect.
Yeah.
That's awesome. Yeah. And I mean, a great definition too, right? Because
Watch.
that's sort of the goal. Like, here we talk a lot about symptoms, and you know, we shouldn't necessarily demonize symptoms, right? Because their body's response, and if you're getting symptoms at the right time, as the correct response, that's sort of a good thing, right? So,
Well, and sure, and the caveat to my little simplistic definition would be, it was sometimes your body functioning as it was designed to do can be harmful. Immune overreactions are an example of that. So yeah, my definition is admittedly simplistic, but I was thinking in terms of,
here it is.
We have this paradigm of like mass vaccination and then pharmaceutical interventions.
Like, what does that even mean, dude? This paradigm of people not perishing before they 50% of us hit puberty. The paradigm, like, what does that even mean? That, yeah, exactly, Rapture. You see that, like, red flag? I was like, what? This guy has no idea what he was talking about. Oh my gosh, it is insane. Yeah, I'm sorry, Mackenzie. I'm sorry.
And it's like the idea of vaccination, for example, is that it's almost as though it's premised on this assumption that, we all require these pharmaceutical interventions for our immune systems to function. And this is
otherwise you interact with viruses and those cause disease. So it's like, you don't require it. We don't require public health. We get to have public health. It's a nice thing to have so that people are living and disease is less. Imagine that. A paradigm of less disease. Oh my, this is
kind of what I was getting at with my simplistic definition, was kind of just to correct that misperception.
Oh, thank you. Thank you.
Yeah, Woo. Sort of like the idea, and correct me if I'm wrong here, but the idea that, we are designed perfectly in a way.
We are designed this is actually where I lost it. I actually lost it on this. One more time.
Correct me if I'm wrong here, but the idea that, we are designed perfectly in a way, right?
Oh,
do you agree with that?
We're designed perfectly, guys. Look at that smarmy little like smirk on the left-hand side too. We are designed perfectly. Like how do you not know about any disease, heritable disease? How do you not know about anything? How have you not encountered a single thing in your life that is an heritable disease? No, Dream. That's honestly a good idea. No, these are vaccines that we have gotten as our community. So we have gotten 1,167 vaccines for our community. In our community, I would love to donate vaccines. That would be an amazing thing. I definitely want to do that. Had a stroke at 31. Perfect. Yeah, but child born with a genetic disease that gives me cancer. Perfect. That's exactly what's supposed to be. Like it's just so like, come on, man. Like, come on.
Yeah, and I think if we give our immune systems, for example, what they need to function, that's you can maintain that state of health, and you can
remain perfect, guys. If you just eat right, you can remain perfect. How can anyone actually believe this? How can anyone understand this? How can someone be this wrong?
Challenged by pathogens and toxins, and your body can function. Your body can detox. It has an immune system. Your immune system can function, and you can maintain a state of health.
That's nice to know.
The problem that I see with the population today and the epidemic proportions of chronic illnesses and so on is that people's bodies are not functioning as they were designed to do. There's just too much environmental
that's it.
exposures that
No, we're
are just
finishing it
ruining
tonight.
people's health.
Oh,
And so
there you go,
the
guys. Oh, it was the medicine all along that was ruining people's health. Oh, oh my gosh, the preventative medicine, guys. That was it. That was ruining people's health. Wow, we were perfect before the medicine. Whoa, wow. Like, guys, aren't there people that don't get the medicine and we know what happens to them already? The people that don't get the medicine when they have those diseases, what happens to you? Anyone? What happens to you when you have a disease, a deadly disease, and you don't get the medicine? God, it's like, oh yeah, you fucking die. Come on. How hard it's just, it's insane that people actually think this.
body has limits in terms of what it's capable of doing, in terms of detoxification, in terms of, you know, the immune system and what it can do. And so I guess the further caveat to my simplistic definition is we need to give the body what it needs to be able to do that function.
Green tea and rosemary tinctures, guys. That's what it is. Oh, dang. The entire time. Just tan, you know, tan your body, get that vitamin D, and, you know,
like proper nourishment.
Proper nourishment.
Yeah, nutrition.
Wow.
You know, avoidance of toxins.
Wow.
And so on. Sure.
Wow.
Exactly.
Oh, wow.
Just out of my curiosity, would you attribute some sort of psychological nutrition in a way, like what you consume on a psychological level? Do you think that has an effect?
What?
In terms of, explain further.
Sure. So say somebody, like an example would be if somebody undergone some sort of severe trauma. You know, if they experienced a traumatic upbringing, maybe physical abuse, and maybe that is a physical infraction, but
Oh, absolutely.
What? Okay. It's not going to I mean, oh my God, this guy's a psychologist. So of course this is what he's like talking about. Nice psychological nutrition. Yeah, exactly. I should have asked my mom when she was dying of cancer, did you just think about being healthy? Duh. Have you ever thought about just not being sad and dying? You should just be happy and alive. Oh, fuck. Fuck, guys. Oh, man. Dude. Man. I'll do that for my next mom. Me, dude. This is it. This might be the last stream. This might actually give me,
Yeah, I mean,
that level.
mental health absolutely ties directly into physical health. Absolutely. There's no question. Yeah, I think so.
Awesome. Great. Appreciate that.
Why did an oncologist think of that? Exactly.
Let's dive in to today's topic, viruses. I'd love to hear.
Fucking idiot.
I think an important place to begin.
I hate these people. I hate these people.
I'd love your input as well. If you think that there's a better starting point.
Wow.
Maybe
Viruses.
we could begin with virus isolation.
Virus
Love to hear your
isolation.
thoughts on that.
Virus, I'm sorry, what?
Isolation.
Isolation.
Oh, wow.
Well, sure. There's this claim that viruses have never been isolated, and they have.
Wow.
And so, you know, when I first started hearing that claim, I you know, as being
Look how bright-eyed, like
positive as I am.
wide-eyed the guy is on the left. He's just like, yeah, and yeah. Some would say that. Yeah, me, I would say that. Yes. What? Oh my God, dude! You just want to like, you just send some virus isolated. Like, what's your PO box? Like, what do you think? How much do you trust the words that you're saying? You know, how about that? How about I just, you know, like, just how much do you trust what you're spewing out?
I would look into them, and I would start investigating those types of claims.
Oh, he's
You
an
know, and one of the
investigative
first claims that I saw.
guys. He's an investigative journalist. This is his job. Okay, there's nobody better in the world to understand what the truth is other than an investigative journalist, and that he has no corporate ties. So he's an investigative, independent journalist. He is the truth sayer. He is the tribunal. Just hand over the keys and please make the decisions for me, Mr. Truth Sayer person. Oh, Kathleen, no, no, no. No, no, no. This is an anti-vax independent journalist going to talk to the guy on the left who doesn't think that viruses exist. And the guy on the right is going to tell him that he's really anti-Semitic. So I think that he thinks that viruses are made by Jewish people. So we're going to see. We're going to see how I'm so surprised. There's going to be, here we go.
Was the claim that like the CDC had admitted that SARS-CoV-2, the coronavirus
guys, he hasn't said that. I don't know. Presumably that's what I'm assuming that he's going to say, but I don't know if that's true. So we'll have to see. We're going to see. Okay. Please be wrong. I love being wrong. I don't want to be right. I said a bad thing. I said a wrong thing purposefully because there's no way that he actually makes that claim, and he's going to make me look like a fool. That's why I said that thing, so that in the future we all look back and we're like, dude, that was so stupid of me. And I'm like, I know I did. So please I said the wrong thing on purpose. We're going to see
causes COVID-19. And let me be very clear about what I mean by that. When I say that the virus causes COVID-19, what I mean is that it's a necessary but insufficient factor in the pathogenesis of COVID-19. So
what? It's a necessary but insufficient what? Because he doesn't think that people are dying from COVID?
The virus causes COVID-19. What I mean is that it's a necessary but insufficient factor in the pathogenesis of COVID-19. So, there's this kind of the debate between germ theory and terrain theory.
There's no debate.
And it's not one or the other, it's both.
Oh my. What? I know what is necessary but insufficient. What in the...
Obviously, host factors are incredibly important, as we just discussed, and I discussed that with my simplistic definition of health.
Both.
So host factors are absolutely important, and to discuss the idea of pathogens like viruses causing illness is not to deny that host factors are determinative.
Determinative. They're determinative. Determinative.
So that's when I say cause, I mean that.
So that's what it is. It's necessary but insufficient. So he's saying that the virus can be there, but it's not making the disease happen. So the virus is real, but like it doesn't actually doesn't do you any harm.
Again, to just quickly summarize that again, it's that SARS-CoV-2 infection or whatever virus infection is a necessary but insufficient factor in the pathogenesis of whatever infectious disease. So anyways, I started, just looking into these claims, like one of the first ones that I encountered was this claim that the CDC had admitted that SARS-CoV-2 had never been isolated, which, you're looking into it and you can see it's demonstrable that claim was false. And so I just kept investigating these claims, and every time I would look at them, I came away with the conclusion that these claims are demonstrably false.
Wow.
Including the claim that the virus.
Yeah, Rapture. He's saying that the virus is so small and that the virus is a real thing, but that the terrain theory, your body, is the ultimate sayer in who lives and dies from the virus-caused disease. That the disease from the virus isn't going to actually kill you if you actually have a fully functioning immune system in a good terrain, which is your body. That's what he's saying.
No virus, not only SARS-CoV-2. I mean, this was kind of the first one that I started seeing a lot. Although I had been aware of these claims that no virus had ever been isolated, like measles and so on. But I never really paid them too much attention before the COVID-19 pandemic. I never really.
They were never popularized like this. It's because the Russian bots hadn't actually disseminated it into everyone's brainstem and installed it like a chip on your NPC brain of like, oh, what thing to believe? What is the cool contrary punk rock position to have, like that I can be like a cool guy and kind of like be seen as a skeptic, and I can like, oh, don't you think? Kind of like, I'm just asking questions, dude.
Dug really deeply into those types of claims until 2020. But again, it's just every time I started digging into what these claims were, and then looking at the sources that were being cited to support those claims, every time the claims are actually contradicted by the cited sources.
Yeah. Wow. I mean, we see that every single time an anti-vaxxer puts, or terrain theory, anyone gives us any piece of information, we just open it up and we read. And reading is the number one disinfectant to conspiracy, because even their own stuff, guys, even their own things that they link us is contrary to what they believe, because they just haven't read it and they have no idea what they're talking about. It's wild. It's actually, it's really interesting.
And so, I find it untenable.
Wow. What's this guy even say to this? Well, that's just like your opinion, you know. I mean, have you ever seen a virus isolated? I mean, like, what do you even say to this?
So when you say isolation, are you referring to, some sort of purity? Is that what you're getting at with that? Or
Bro, like, I don't. Do they even understand what a virus is? They understand that it's not a free living organism? Like, do they get that? Do they understand that, like, what he's talking about is that it's going to be, you just have, like, liver, kidney cells, and then you have the virus. You don't have the virus by itself. That's what it's actually saying. Hi, Nightflyer. Oh, no, Stealth. Oh, no. Well, we're your new friends now. You're welcome to stay here. You're welcome to stay here. Oh, yeah, exactly, Dream. What are the viruses, the friends we made along the way?
like, because I guess my understanding of isolation virology is,
Wow.
Isolating the virus out of a host into a cell culture. Is that
Yeah.
what you're referring to, or?
It's usually how it works.
Yeah, cell culture is considered the gold standard as it's described in the literature of virus isolation. Yeah.
Okay.
Exactly, man.
But cell culture alone is not the only means that scientists have at their disposal to be able to characterize, to identify and characterize viruses. There's also whole genome sequencing is another really important example.
Shit. Look at this guy.
So it's not just,
we got a brainiac over here. What are you trying to impress us? What do these words mean? How old is this dude? Like, older than 16, so he has no excuse for this crap. It's insane.
The cell culture, that is evidence of the existence of viruses. But that is what's considered the gold standard for isolation.
I mean, he is right, and he is right, guys. I mean, it's like isolation, but then on the isolation part, every step of the way, you could be doing sequencing and PCR and 16S ribosomal sequencing, and you know exactly what it is, what organisms are present. You can do it with and without your control. It's really easy, really, really not hard. Done. Yeah, he is anti-vax. He is anti-vax.
Got it. Okay.
Got it. Okay. Look at that face. I can't. There's no way this is another hour. Guys, it's another hour and 4 minutes. This is unbelievable to me. Look at that guy's face. Like, he's just like so, like, dang.
And I think that's one of the, where the argument, where people really get stuck on the argument there that that's not isolation, right? Is where when we start discussing like Rivers' review of Koch's postulates, right? Like
my God.
within it says there needs to be some sort of isolation, some sort of purity introduced to a host to
That
prove
is
that that is the causative agent in some sort of infectious disease. I think
what?
that's largely where
Oh, Kathleen, he has none. He flunked out of biochem and then went back for his psychology master's. So he has a psychology degree, and now he's you know, yeah. I'm sorry, hon. This is gonna be all night. I'm sorry. You
that
just
is applicable to this argument. The other one is that you just brought up is
again, guys. Again, like he knows more psychology than I do, right? I wouldn't just be going in there and be like downplaying the entire role of the brain and how psychology works. You know what I mean? Like, imagine that. And being like, I don't actually believe that psychology and the brain means anything in how you react and who you are as a person. And they're like, what? What are you talking about? And you're like, no, that doesn't have anything to do with it. It's about your toenails, actually. And they're like, whoa, have you ever isolated a personality from a brain before? I think that both of them are probably. The one on the right is an anti-vaxxer, confirmed.
genomics, right?
Yo,
Isolation of, and I would love to know if you agree with this. I don't necessarily think that isolation of viral genetic material, especially of a single species of virus, is a possibility.
Like, do we need to fucking show you the nucleotides? Do we need to show you like the millions of reads of ACUs and Gs? Like what could you possibly need in order to like have this thing be this proven that's so easily? They literally have this. They have, God. So okay. There's RNA contamination of viral of sequencing that does exist, guys. There are literal like RNA contamination, that they can find, and they can find it. They can sequence the RNA in DNA sampling errors, and we know like a lot more. They actually ran a huge sequencing DeepSeek on all this like extra DNA that they had because they were junk reads before, right? They didn't map on anything, so they just threw them out, and they were like, wow, maybe there's like viral genomes in there, and they did it, and they found like a bunch of viruses and stuff. It was really freaking cool, man. So it's like you don't need in the next generation sequencing technology. You don't need a single viral particulate or a single viral isolate in order to do the whole genome sequencing of the virus. That's the whole point. Like, welcome to the 2010s, dude. He's talking about Sanger sequencing, which is the 1970s and 80s and 90s, dude. That's completely different. We can sequence entire microbial communities now: viruses, microbes, bacteria, yeast, algae, all together in a community. We sequence it all and we figure out all their genomes. Like, that's where we're actually at. This guy thinks that we're back in like the 1700s still, dude. Like, come on, this is so ridiculous.
Well, there's actually, this is another argument that I've seen repeatedly made, is that
Sure.
that
They're missing a few updates, exactly.
That you can't do whole genome sequencing of a virus when that genetic material is mixed together with a bunch of genetic material from other things.
Yeah, Maria, they do. Yeah, and Maria, they're all separate too. They're all distinctly evolved separately. That's what's the craziest thing. So, you know how like eukaryotes all have like the same similar function and stuff like that, like where they'll have, their morphology will all be similar, and we've like all eukaryotes have like, we have a very similar body and genetics and stuff, like 16S ribosomal sequencing, all that kind of stuff. We've actually had, it's very life like is very related to one another. Viruses don't have that. There is no single piece of viral genome that is contiguous to every single virus. So our current hypothesis is that separate viral clades or whatever they're called. I mean, domains like evolved actually separately. Exactly, Maria. How insane is that? It is unfucking believable. It is amazing. Or we don't know anything, and these guys are the ones that are right, and it's just, you know, we're watching both sides, guys. We're watching both sides.
And that's actually just plain incorrect. They actually have what's called, metagenomic sequencing.
Look at that.
And for an example of the utility of metagenomic sequencing is scientists, doing this type of sequencing on the gut microbiome to be able to identify hundreds of thousands of different types of viruses that reside in the gut. Mostly, these would be viruses that infect not your human cells, but the bacterial microbiome. So viruses that infect
Look at this
bacteria are
give it up to this guy.
What's the word? What are they called? Macrophages.
Okay.
And so, they have done metagenomic sequencing, for that purpose. For example, there's one example that leaps to mind where, that it demonstrates the utility of metagenomics for determining the whole genome sequences of a multitude of viruses, without having to first isolate each one individually.
Yes, exactly.
So you can do metagenomic sequencing without actually isolating an individual virus.
Yes, he's exactly
this is
right.
It's just a big misconception, and there's a lot of falsehoods about that. People claiming that that's not possible, but
he's right.
Scientists do it.
Yes, you do.
I think where I would take that is not necessarily that it's not possible to run metagenomic experiments. I think I would question, the validity of the results of the metagenomic process. I think that's where I would
What are you talking about?
Like, that's where I would get stuck on, because Sanger sequencing is the kind of the gold standard of
he literally explained it to him what the current state of technology is, and he's like, yeah, but like, it's like, it's like getting a Formula One 2025 car and you didn't win the race. And they're like, well, what happened to your car? What do you think, like, what is the problem? Why didn't you, like, go fast enough? And he's like, well, the 1978 Pinto had a lot of problems with the rear engine. Why don't we, do something like that? Why don't we go back to what happened and fix us then, and then we can just apply that now? It's like, what are you talking about? What can you just stay here as if we haven't, how you don't know anything to know that you don't know. This is the Dunning-Kruger effect, ladies and gentlemen. This is the Dunning-Kruger effect, exactly what they're talking about. People that know the least are the most confident in their knowledge. And he's literally writing off all of next generation sequencing because Sanger sequencing is actually the gold standard. Good to know. That's good to me. That's good to know. Dang.
Genomic assembly, right? Or, genomic sequencing.
No. It's exactly
wrong.
wrong.
And purity would be necessary to run Sanger. Like, I don't think you can run Sanger with viruses.
A fucking course you can. You can run Sanger with viruses. Oh my god. You just amplify that part of their genome and then you put it into Sanger sequencing, guys. Like, it doesn't, you just amplify their part of their genome and then you sequence that. You sequence that, you use 16S ribosomal sequencing, you just amplify that portion of their genome and then you see what it is and then you sequence that and then that's what it is. This is so simple, man. Come on.
I'm not sure about Sanger sequencing in terms of whether Dr. Singhong Lee would be the expert on that and would be able to answer that question. But
Sure. Look how happy he is. Yeah, you didn't think of that, did you? You didn't think of that, did you? You didn't think of that. You didn't think of that, huh?
In terms of whether they do metagenomic sequencing with that technology, I'm not sure about that. I can't speak to that exactly.
It's because it's fundamental to the technology and what it is. Basically, because of next generation sequencing, you can add adapters to the ends, and you can have unique barcodes on every single piece, and that is what allows you to have millions of pieces of DNA that is effectively unique, and that's how you actually know what is what in that whole thing, because they're all different, as opposed to just sequencing the same piece of DNA 1,000 times, right? So that's the whole point. And Sanger sequencing is a whole different technology. Basically, as the amplicon is degraded, it has a fluorophore going on. But the other way with sequencing by synthesis is completely different. The fluorophore, as it's being assimilated and onto the piece of DNA, that's when the fluorophore is coming. So then you can do that, and then you can decouple the DNA, I can unzip it, and then you can sequence the other side too. So you get parity. So you actually get double the amount, and you can understand what's actually going on each side, and then that confirms it. It's incredible. It's incredible. Oh.
But I think one of the areas of confusion with, accepting the results of metagenomic sequencing is just kind of a misunderstanding of how whole genome sequencing works. Mm-hmm. And again, I'm not a scientist. I'm not an expert on any of these things. I just know what I know from researching these claims about the non-existence of viruses and just like digging into those things and spending a really extraordinary amount of time on it.
None of them, Ghostbusters, none of them. They're both idiots.
So one of
Look how happy he is. Look how happy the guy on the left is. He's like, yeah, I did it. He researched. Yeah, Heather, exactly.
One of the claims, that is kind of made is that, well, one of the claims that I saw early on was that, that you can't.
You talk a lot of BS with little results. What does your spouse, child, and grandchildren think? Well, your mom gave me a 5-star review last night, so, pretty good, I think.
Can't do whole genome sequencing without a reference genome, which is kind of a logical impossibility. If you had to first have a sequenced genome to be able to sequence subsequent genomes, to subsequently sequence other genomes, well, this is so cringe. Then how did you sequence the reference genome in the first place, right? And so there's something called de novo sequencing, where it's like the very first time, where you sequence something for the first time, and then after that, then you can use that sequence as what's called a reference genome for subsequent, types of genomic sequencing. But there's de novo sequencing, and metagenomic sequencing is a type of de novo sequencing where, again, you don't even need to isolate a specific, an individual virus to be able to do that.
I'm sorry, Kung Fu. I'm sorry.
Type of sequencing.
What's up, MT?
You can sequence a multitude of viruses or other pathogens directly from a patient sample, for example.
Yes.
Using this technology. And so another kind of, I think, misunderstanding is how
like Jorge, what's up, dude? Dude, it'd be like, nah, I don't think that the internet actually you can like use it to talk to people. And you're like, huh? Like we're doing that right now. And you're like, nah, I don't think it actually works. I don't think the internet actually does anything. And the guy's like, what? It's like, bro, we're doing that right now. Like we're doing that right. Do you want me to get out a USB and show you what freaking sequencing looks like? Like, what are you talking about?
How it works in terms of what it is. And so it's not the case that, as I've seen people claim, that, they just kind of randomly assemble the genome from these little snippets.
Loser says what? I love it. Good. That was a good comeback, dude. That was really good. That was amazing.
Bits of genetic information. It's not randomly assembled any more than you randomly assemble a puzzle when you're putting together a puzzle. I mean, the pieces lock together in one way. This is why the puzzle analogy is of course imperfect, but it's useful to kind of simplify the concept of that. The different sequences that they assemble with this technology. That there's overlaps in the sequences, and so they can assemble them in the way that they fit together.
Yeah, dude, it's a puzzle box. Okay, you got a puzzle piece, you have 1,000 pieces of puzzles, and like you have a puzzle box. With the reference genome, you can look and be like, oh, okay, this is a wing piece. This goes right here. This goes right here. This is a corner piece. Okay, this is here. If you don't have a reference genome, then it is extremely hard. But you still have like, hey, this is an organism that's evolved on Earth. Wow, brain buster here. We have things that we can like look up, and we can blast portions of the genome. We can see what it's called an open reading frame, what this ORF will make, what the protein and what the coding sequence is, which is what makes the protein itself. Like, wow, this actually aligns with this. These things align with this. We're not just like, this looks good, and you're doing it by computers, so you can like reference this stuff. So because these things are gigantic. These are, tons and millions of pieces of nucleotides,
and so they do fit together in a specific way. And you can't just like take these snippets of genetic sequences and just randomly assemble them to just make up.
Man, Jorge, I don't know. I don't know. That's a good question. Dude, honestly, dude, I am like more impressive by the 99.9% of microbes that we haven't sequenced or cultured. Like, I think that's I think we have like alien life forms on Earth that we have not even explored, bro. Like different new proteins, new metabolic chains, new gatekeeping mechanisms in order to make more advanced proteins. Like, it's just, I just, my mind gets blown at like what's at the bottom of the Marianas Trench. What organisms and microbes are living inside the crust of, beneath the crust of the Earth, underneath, our oceans, that kind of stuff. And they have sampled some of that stuff, and it is really cool. It is very, very cool.
Whatever. It just doesn't work that way. And this is one of the biggest misconceptions that I've seen out there in terms of how it works is the claim that they just kind of randomly assemble this genetic material to come up with whatever they want. And it's just not how the technology.
Like, why would you do that? Like, why, if you're a scientist and you've done this experiment, okay, and your publication rides on this data, like, and you need to show that your treatment actually worked. You know what I mean? Like, you got to show through your sequencing that this actually worked. So do you think that you're going to be able to show one is better than the other if you just randomly assimilated them all? Like, no. There's going to, more than likely, because it's a random assortment, there's going to be no difference. Because all the differences have been, what? Randomized and averaged. So anything that you use for sequencing with this brain dead method wouldn't show any significance. Like, it's just, these people aren't serious. These are not serious people.
Operates.
Interesting. Appreciate
interesting.
that.
Interesting.
On the topic, the idea, the concept of variance comes up in my mind.
Oh God, it's gonna be an evolution question.
Because when we're talking about this de novo sequencing, I would probably agree that having a reference genome would be helpful in like validating this. Of course, that just makes good sense. Is it absolutely necessary? You're arguing against that, which I can appreciate as well. When it comes to that de novo assembly, we see the variance. That's one of the things, the variations in these genetic sequences, slight variations. I'd love to hear your thoughts on that, because to me, in my sort of read of it, is that could be indicative of just an error in the assembly.
Yes.
Like
yes.
because, and maybe you could speak to this as well. I know I'm sorry to throw a few things at you here, but um, in the assembly during that process, I was understood that these um, platforms, like these softwares that put all of these genetic sequences together, that sort of build the puzzle, is probability based. Um, and that it does synthesize a mass amount of sequences. Like when you run it through these systems, you get sort of a ton of different sequences, and you may get 2 or 3 sequences that match, the
Yeah.
length that you're sort of searching for.
No.
So if it's a certain amount of base pairs, if it's
Millions.
30,000 base pairs, you have 2 sequences that were synthesized. So there's 2 variations of this virus. Um, that was sort of my understanding. Maybe you sort of just did clear that up, but if you want to clear that up anymore, I'd appreciate your thoughts on that.
Oh my God. My God. Okay. So there's like the base calling. All right. If you have a chip, you literally have a chip on this thing, and there's like lines, right? There's lines. Think of it like it's your monitor, like literally like the computer monitor that you are staring at me on right now. Think about if these LED panels are lines of DNA, and that's literally what the sequencing by synthesis Illumina technology has. These like little things that literally go on, and that's what binds to the DNA. So it looks exactly like this. Okay. If you have a huge swath of yellow, right, it's going to be hard to see a little red dot. We know that, right? So this is called a miscall on there. So what you do, and how you know that limitation in the technology, what you do is you basically phase the sequencing. You don't line them up like this or like this, okay? You stagger it a little bit so that, if you have, and we're talking not 2, brother, we're talking about millions of pieces of DNA. 2 pieces of DNA, dude, come on. You stagger it so that the sequences aren't going to be like that. It's going to be red, yellow, green, blue, everywhere. And that allows more definition for the camera to actually see the variants and to actually call each one correctly. All right. In the preparation and what we were talking about before, the sequencing by synthesis, it calls both sides. So like the fact that there is, going to be some error in both of the reads on every, and it's very, 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 very unlikely, insanely unlikely. There could be an error in creating the library, right? Especially when you're using PCR. PCR has an error rate in it, was 1 in a billion, right? But what if you got super unlucky and you got that PCR error on the first amplification, right? So literally on the first one, well, guess what? Your entire population is going to have have those different variants, right? But guess what, guys? Oh no, what do we do? What do we do? That's why we run 2 samples. We run 2 or 3 samples to make sure that there is a replicate, and that you actually know that the other one is legit. That we know this. That we spend our entire lives making sure that we aren't tripped up by this, because we're spending $10,000 on a sequencing run. So we want to make sure that isn't a computer error or an instrument error or a PCR error that's going to hose the entire experiment. It's like that is what we spend our time doing. Welcome to science. Welcome to expertise. Welcome to what our job is. We actually spend our life actually trying to make this better. That is the whole point of what we do, man.
Oh. Well, sure. When they speak of viral variants, they viruses mutate and evolve, right? So the original Wuhan strain of SARS-CoV-2 is different from the subsequent strains that were. Or I shouldn't use.
He didn't say lab leak or natural zoonotic spread there. Did you see that? I guarantee. I'd bet my hat that this guy is a lab leaker.
The word strain, because scientists differentiate between strain and variant, but
oh my God,
you get my point. That the variants that were subsequently circulating, displaced the original variant.
Huh?
and so it's as the virus moves through the population, as it infects people within individuals, it can evolve and mutate, and then it spreads to someone else, and there's further evolution and mutation
Okay.
of the virus. So this is what is meant by variants: is just they're mutations of the virus. And if those
Oh
mutations
moly.
become significant enough, then they could describe it as as a new strain. But the variants are kind of considered the same strain of virus. It's just that there are genetic mutations there that may be significantly meaningful and may not be. But typically they name the variants of SARS-CoV-2, for example, based on what they consider to be significant um, evolutions in the viral genome.
Oh my
Omicron,
gosh.
for example, having a number of significant mutations in the spike protein that made it that enabled it to escape the antibodies from prior infections and from vaccinations to a significant degree.
Okay.
So that's an example of
Okay.
what they refer to as variants.
He doesn't know the sequencing
But
technology.
you're right that there can be errors, like in reads and things
Oh
like
my
this. But
god.
there's also
Idiot.
you can run sequencing multiple times.
Yeah.
Scientists around the world, on a regular basis, you sequence genomes of the virus, and so there's this kind of this redundancy and replication among scientists of the genomic sequences. And so when you there's a very kind of insignificant I would say basis to say that that the outcome of these these computations, which, because of course whole genome sequencing involves computation, because as you explained, I mean, it's something that the human brain can't do. It requires a
You
computer.
definitely can.
So they have
I
software.
mean, you definitely can. I mean, like, I do it. Like literally, you get sequencing back or Sanger sequencing or readily or something, and you just kind of like look, at the sequence that you want. You just Control F, find the sequence that you want, and see if there was an indel, an insertion, or deletion at that point. You don't need necessarily like, you can do it, but I mean, it well enough as a scientist in order to go through that. But like, if you're, crunching sequencing from entire population, and you're assembling contigs, which is the assemblies of the DNA, and then you're trying to blast them, which is searching the database for it. Like, you can't just do that readily. That you could theoretically. I mean, all of the DNA sequences are just in text files, guys. I mean, it's literally a text file. You still literally can Control F and try to find it. I mean, it's literal. I mean, that is what it is. So it's not like some secret, Boolean string that you don't understand. It's like, no, it's literally text strings, guys. There's literal just like ACTs and Gs in a you can open it in Notepad. It's like, come on, dude.
For that, and so it's not the case that you're just getting these errors and it's just spitting out whatever. It's yes, it's true that you can have read errors and things like that, or assembly errors. But at the same time, when scientists are replicating the process over and over and over again on a daily basis, then there's kind of a limited room. There's kind of limited room to say that, well, we don't accept the outcome of the genomic sequencing, right? As many people do, and they just dismiss whole genome sequencing altogether. And I just find that just untenable and unsustainable.
Okay. It seems as though this conversation is quite far away from life. We're in the computer with this. There may be, this is the interesting thing. You know, if we accept that the genomics, the metagenomics, is an accurate representation of what is in some sort of sample biologically, um, it still seems as though it's quite far away to make claims um, about these little particles, that where
They're so little, you can't even see them. just can't. Have you ever seen a nucleotide? Have you ever seen one? Have you ever seen one? Have you ever seen one? What you're talking about? It's like, yeah, dude, we work on inference a lot. That's the whole idea. Like, did you see the murderer? No, but the police can like go through and figure out who it was based off of data. And like, you know what I mean? Like that's how analysis works. That's how people do the work. That's the whole point is.
The isolation process is not really some purity, especially when it comes to making claims about, their pathogenicity. You know what I mean? Like if it's going to cause disease.
Like what is he expecting to see? Like what is the thing that he expects just to be like, oh, guys, whoa, oh shit, oh yeah, I got one. I got the virus right here. Oh shit, oh, I got it. It's right here. Whoa, oh, virus. I got the virus right here. Oh, I isolated it. Oh, jeez. Oh my God. Oh, that was close. It almost got away. That virus, it almost got away. Woo! Wow. Wow. That was close. Jeez. What's the process he's expecting? What is he expecting to see? Like what is he expecting to get? I don't understand. Like what does he want from us? Genuinely. Like what does he want? Yeah, exactly. It doesn't work like that. It doesn't. It's actually very surprising. And this is my new. I've been trying to do that whole. Everyone does like the thing on the thing. You know what I mean, with the mic on the thing. So I'm gonna do a pipette. I got a pipette for my thing. Isn't that good? Isn't that good? Pretty good. Pretty good. Like it. MT, what's up, dude?
Bro, did you?
What was that? No, I got this for $20 off of Amazon. This is a cheapo, awful pipette, bro. I would never use this thing. This thing is garbage. Oh, I can't hear you. Your mic is going in and out. Try again. Yeah,
Can you see me?
There we go. Yeah, there you go. Oh, look at you, dude. Look at you. Oh, those are the Eppendorf Research Pros, dude. That's solid. You got some good stuff. You got the good.
I wasn't trying to flex on you, bro.
Dude,
it's the Research Plus.
Yeah. I know. I saw. Those are my favorites.
I wasn't trying to flex on you, bro.
Those are my favorites. Yeah, they are. I love the Research Pros, dude.
Work for a Fortune 500 company, they don't spare no expense.
Dude, especially the multi-channel ones, those are really nice. Those are my faves.
Yeah, I'm confused by this. I was watching a little bit of this video.
Mm-hmm.
I'm confused. Isn't that like your boy up there on the right side? Is that your boy?
No. So this is someone came up and was had some questions yesterday, and then they got referred to this guy Beyond Terrain as this is the YouTuber on the left, and then he was like, oh, he even has people that he disagrees with on, and this was the one that he listened to that had the contrary opinion person on, but the guy's an anti-vaxxer, a journalist, not a scientist. Yeah. So he doesn't really understand what's going on.
Yeah, I was confused by that. And then, I find too, because the dude on the right, he is a scientist, right? Like he.
No, he's a journalist.
No. Okay.
Yeah.
Okay. 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 That's what I was confused by, because like because he was saying shit. And I'm like, this doesn't actually make any sense. And
it's
because I was just listening, I'm walking around, and I'm like, what the hell's going on?
Yeah, exactly.
Okay, so it's a very disingenuous argument between two non-scientists
about virology and about microbiology and the fact that like microbes actually exist. The guy on the left does not believe that viruses exist. He doesn't believe that.
Okay.
We don't know his vaccine views, but he's probably an anti-vaxxer. Guy on the right is a fervent anti-vaxxer, and that he believes at least that viruses are real. He just doesn't believe that they like actually can kill you or cause a huge
so
part.
What they have done is they have created cyclical anti-vax content. So it doesn't actually matter that it's fake. It's just for them to collaborate on their common ground and make content.
Exactly, dude. Yeah, this whole channel that he has, bro, it's unbelievable. Like there's so many interviews that he does that are just with all these like doctors and stuff. I mean, this isn't going to be
How many subs does he have?
4,000. I mean, it's not like a huge YouTuber, you know what I mean? But
yeah,
but his views, I mean, we're talking 1,000 views, 1,000 views, 3,000 views.
Yeah.
I mean, it's like, and he's been going for a while. How long is the channel? 2 years. So. Yeah, the most popular one that he has is 3.1K. Yeah, lots of this nonsense. The immune here we go on the right. The immune system doesn't exist. Viruses do not exist.
Yeah,
it was really interesting. He was talking about how DNA isn't for heredity, and I was like, wow, dude, he's like talking about Lysenkoism. I was like, that's like wild, dude. Oh yeah, there are no contagious diseases, like that kind of stuff. You're just like, oh my god.
Part of the problem, honestly, is that we engage with these people in the first place. I mean, I know that dude on the right actually is not a scientist or whatever, in that context, but like, I don't know, like there comes a point where it's like doing more harm than good to be someone of actual credibility and stature and like speaking with that person. It makes more sense, I guess, in your setting to have people come to your platform who don't believe those things than to go to some, you know what I mean? Like,
yeah, but the problem is like I had someone come in and they say and argue that this is, yeah, exactly. Yeah. So it's not just like me, and it's not about like, platforming him, and it's more of like people have similar ideas. Even if he doesn't have a big platform, he's saying things that people have questions on. So that's the thing.
I'm not even gonna lie, it really actually doesn't even come down to platforming. Like I'm just thinking about the sheer, not only like the ROI, but just like the productivity. Like it's actually like completely just counterproductive for someone like that who is making money to do that. Like it's easier to engage. Like that person who recommended it to you, like he's obviously not the person who's recommending these things, they're not firm in any sort of real beliefs. They're kind of just getting fed stuff.
Yeah.
What platform? YouTube, Spotify, of course.
Yeah, he had a Substack. Shoot, is it the guy on the right or one of them that's a $40 a month Substack, and he has 10,000 followers on Substack? I'm like, oh my gosh, dude.
Yeah, but they can't all be paid.
Yeah. For real. But I'm just saying, like, it's
crazy. I just saw someone the other day that they had a $30 a month, and I'm like, what the hell are you doing? Like, and they're not leftist, but they're like, I guess it's because they're liberal. I'm like, bro, what the fuck are you doing with a $30 a month Substack? Like, my God, dude. You think you a fucking newspaper? Like,
Just trying to get those Oilers, man. I guess getting the Sharks or the Whales.
But yeah, no, that's crazy. The people, doing these things, and I see now because the way that you're kind of coordinating your Discord and whatnot,
Mhm.
you're kind of gonna try to make it a science shop, like a science fact shop in a way, right? Like you're trying to court other scientists in there and stuff.
Or what, with my Discord or my channel?
You're just, well, I guess both, but like your Discord, like your like your little ammo station.
Yeah, I mean,
where you're putting your like your research and stuff and like the graphs and just building up all of the repertoire to make these arguments.
yeah. I did not see that Kung Fu. There's parts of that like for the stream that I wanted to, but it really is just organizing. I use it for like the Discord in general. So for the stream, like data tables and papers and stuff are good, but then also it's like for our like nonprofit or is all organized there and all that kind of stuff. So I just see it as kind of like the hub. Discord is just the hub for everything and all communication.
No, yeah, that's the way I'm seeing it too. And I'm trying to figure out a way to natively use it for like other people. So, I guess shifting to like the CEO persona in ways, it started to make me think about big picture and just how to like ramp up like super quick. And so it's like, well, I already have these platforms. I might as well just have a bunch of people that I trust like using my platforms, and I'm just focused on like different things like I don't know, not saying like I'm just gonna not make content, because of course I have to like be out here and make pods and whatnot, but just in terms of like like collecting all that stuff, so I'm like, man, I already got with you, and I feel like I have 5 or 6 PhDs just in my Discord already. So it's like, okay, well, if I can put together like 20 PhDs real quick, now we got a freaking science corner. Now we have a place generating factual information consistently that we'll always have to tap into to be able to spread, appropriate information,
Yeah. Yeah, absolutely. Yeah, I think that's a good idea. Like I originally, and I do think that there is a need for like infographics and explainers for a lot of stuff like that, where we could create things that are easily shareable on social media, but that actually are easy, that convey information, that have sources that can be fact checked, that kind of stuff. And then, that's just kind of like, I did the one for Lab Leak, back in the day, like years ago. And so that's what but I was thinking about doing one for HR, thinking about doing one for like vaccines and, misinformation too, but just haven't, that's a tougher one for me. Cause it's just like, the anti-vax case is, I feel like more an appeal to it's the anti-vax stuff is more like, don't tell me what to do kind of stuff, and it's not gonna, they don't care about that. So
no, of course, but one thing that I've noticed, so you actually mentioned it earlier. I feel like it had to have been you. I feel like I was watching you somewhere else, and you brought up, how everyone has to be an expert because of the way that the algorithm works, and we just try to get the engagement and stuff like that.
Yeah.
And it made me realize, scientists, especially PhD scientists, are actually at a huge disadvantage in communicating effectively, like online, because they might not actually want to be delving into too much stuff because they actually do have an expertise and credibility. And so if they go a little too far in too many places, it could actually, like, in some ways maybe hurt their credibility, or I don't necessarily know how y'all view those types of things.
Sure.
But it is one thing I noticed is that a lot of scientists with their content, if they make content, is very niche.
Yeah.
Which I kind of like. You're pretty wide in terms of
I try to do that.
What you're bringing to the table.
Yeah, I try to do that specifically. Like at least with I'm going to like, I mean, anything outside, even in microbiology. I mean, I'm just learning here. I'm learning on stream with you guys, basically. So we're going to original documents, reading papers, that kind of stuff. Like I don't know that much even about microbiology, to be honest with you. So, and I have a
Right.
PhD in it, and it's like because the knowledge and the is so deep. There's no way, man. I just skimmed the surface, you know. And I'm basically only a world expert on like what I did personally. So like using nanotechnology to deliver DNA to algae. Like, as long as we're in that lane, I'm gonna be okay. But anything outside of that, I'm just kind of like learning here with you guys. So it's
Yeah, no, that's just one of the ideas that came to me because I happened to see I saw a PhD person join my Discord today, and so I just went go look through the members, and I was like, wow, like, and I happened to add one to our group chat, Dr. Kamika.
Oh, nice, yeah.
And you know, that's what made me realize, like, man, I'm actually natively, like, really organically just cultivating like such high level, you know, scientific people. Oh, and Dr. Taylor, PhD, I just had him on the pod last week.
Nice.
You know, so yeah, I feel like that's something that I kind of want to put a little energy into.
Nice, man.
And you know, in no way I'm trying to like co-opt you or anything. I mean,
No, we're doing it
it's gonna be
together.
a whole thing.
We're doing it together.
That's exactly it.
Yeah,
We're doing it together, and so that's one of the aspects of maybe like the cross collaborations and whatnot is just keeping me in the flow of your scientific information and just, you know, helping to cultivate the scientific community that, you know, I kind of envision as really a core, a very true, like fundamental core piece of a real propaganda machine. Like, because we're not lying to people. I know it's called a propaganda factory, but like,
yeah.
I'm not going to sit here and waste my time just lying to people.
Yeah. Yeah, exactly. No, I think that I think that like the, you know, science and what's happening now in science is so like awesome that it really does feel like outer worldly science fiction. You know, I've thought about that a lot where it's like you don't even have to, people are like, oh, what about UFO technology and stuff like that? And, you know, maybe how, you know, what would that be like, you know? And I'm like, bro, like I could explain to you right now what we do in science and that would seem to be like UFO technology, right? So I think using that and kind of showing people that this stuff exists and we are already doing this. So like, do you really not think that we could do this simple thing? You know what I mean? Like we could, it's just actually wild, like how advanced the science already is. So the idea that we couldn't like do this really, really simple process is, you know, pretty arbitrary. Yeah, Laughter and Light's in here. She's awesome. She's a fan and she watches a lot. So yeah, dude, Laughter and Light. I'd seen somewhere she mentioned her research and I looked it up and she's like legit. I honestly think Laughter and Light probably might be the most credentialed science communicator I've ever seen as a scientist. She's like she's up there, man. She's really good. So
yeah, that's my immediate goal is to try to put together like a small team real quick of PhD influencers and then leverage that to try to recruit more and get to like a critical mass. Or even if they aren't influencers, but you know, nowadays they seem like, well, you know, smart people got opinions.
yeah, exactly.
Like you said, like just that science communicating. I mean, it's very crucial, and we find ourselves right now because of the lack of science communication, and in some ways, it's partly because of the way that it's developed. I'm not even going to lie to you, the way that the university systems work and the way that the knowledge, it's not really democratized, but with the content creation game, you know, they kind of opened it up for a lot of people to have that exposure, you know, and I feel like that's part of what you're doing, like you said.
Yeah, Stealthcast. I am the new Bill Nye. That's the whole idea. I am the science correspondent for like streamers and stuff. That's literally how I pitch my what's going on. So I love Bill Nye. I love Bill Nye the Science Guy. I would love to have like, you know, in the future, some type of like, that would be a cool thing to have like an actual budget for a show, like where you're actually just debunking stuff and actually doing like experiments like they do. Like, honestly, guys, you should look back and look at Bill Nye the Science Guy episodes, I think it was genuinely revolutionary. Like, you should watch it, and like the fast cuts, like the editing that he does, the joke breaks, it's part like comedy skit, part science, a bunch of practical experiments, but also the production and the editing was next level, bro. We didn't see things like that for 20 years. In shows, it was truly innovative.
I think that you would be surprised with the production value that we'll be able to probably see, um, over the next few months. I'm not. It's just with the momentum that like we have with putting all these awesome people together. Like, there's no way that we're not going to be able to just keep up in the production, up in the value, up in the collaboration. Because I had the idea the other day, because you have all these content creators, and I was like, we need to come up with a way for all of us to like, we need like a content bank.
Yeah.
So because we might have overflows of content or whatever, or we can even like plan productions of content, and it's like we're all producing for the same piece of content, and then someone's going to edit that, and we have some sort of collective page or just something that we all agree that we're donating to as our collective war chest.
Yeah, Dr. Joe did that with
that
a science group. Like, it was like 8 of us, 6 to 8 of us, and he asked for a 30-second thing on something, and mine was like debunking aluminum and vaccines. So like that was like made like a little 30-second thing, and then he like cut them all together and made like one super cut and stuff, and it was like, yeah, it was cool. There's a lot of people on it.
That's exactly it. And I feel like we could have creator teams doing, maybe, I don't know, because some people's content creation is different, but like we could have daily cuts, weekly cuts, whatever. Like, there's so much availability we have to like put together short form content to create more higher production, higher quality long form content, because we're all generating these things. And I'm thinking back to the times in which I used to mindlessly sit there on YouTube and whatnot and watch compilations. And I guess in some way it's the same thing, but it's producing the compilations in real time. So we're collectively thinking, okay, we're all going to make this type of content. We're going to try to make it in a tight format, or we're going to do piggyback segments. Like, this is your segment, this is my segment, boom, boom, boom. This is our show for today, and we're trying to have something out like every day, 7, 8 o'clock. That the whatever is dropping, and it's
Yeah, dude. I mean, that's like, I mean, because you can even have people like try to do their own stuff, like publish their shorts on their own channel, and then just send in the short to like a longer compilation on YouTube or something.
Yes,
so then
that's
it's
exactly
like,
it.
it's like
double monetization.
It would be like Adult Swim, or something like that,
Yes,
where you're just like watching it and you're like, oh shit, yeah. So it's like you could be like scrolling through shorts, but you don't know what the next short is. But this way, it's kind of like all the same, idea.
The same flow.
Yeah, exactly. Yeah.
Or even, like our production for the day. Like, let's say, for example, there's protests going on, and so it's like, okay, team, go correspond, or something happens in an area, and it's like, I'm gonna go pull up. You know what I mean? Like, you have to think newsrooms have essentially been gutted, and scientists are in a unique position to make up for journalism, but they can't do it by themselves. And so if you can build these teams and you can build these, think about warfare. Let's go circle back to Napoleon, that guy who, that freaking crazy dude who was up there.
Yeah.
But if we think about the way that Napoleon revolutionized the way that modern armies are designed, and that almost everything is included in a troop now, versus back then it used to be the artillery or the whatever. It's like now you have a small unit that can operate independently. We need to replicate that and create that for creator systems. And it's going to be kind of hard because you can't just pick and pluck everybody, but creating organic relationships of people of diverse talents, diverse backgrounds, and just flooding the zone. I mean, that's the whole key.
I think people are like looking for that stuff too. I mean, everyone's trying to look for an idea. Like I just started doing the duets or stitches. I just learned that tech. I was like, how do I green screen and then cut? And then I was like, what the fuck? I felt such like a boomer, because it's like there is like a learning curve with like using the TikTok app editing and stuff. So it's like even having someone to talk shop with and like ask and make better videos and or different content ideas and stuff like that, or what I mean. So yeah, I think that there's tons of space, man. I feel like organization is just hitting. Like I really do feel like Discord is revolutionized the community aspect of it. It really feels like a big community in there, and I feel like there's like so much untapped potential in like actually organizing people, as in like to do the political actions, and 501c4s and actually creating something for a nonprofit, and then also like the creation of that content. I don't think that's been utilized at all. I think that there's tons of innovation there.
I agree. And any of the content that because you have to think. I'm not even going to lie to anyone. When I'm thinking about this and I explain it, it does somewhat sound like MLM-y, but like MLMs are based around like a product and people making money. And then, social media teams or whatever, like those things already exist too. But again, it's not centered around anything realistic, And so I think that it's kind of the exact same thing. I mean, this is on a very fundamental level, somewhat a multi-level marketing thing, but it's marketing like the right things. Like,
no, it's like, I mean, it's basically like a work from home streamer house.
yeah.
Yes. So
that's exactly it. Antifa is a work from home streamer house.
There you go. Exactly. Perfect. Yeah. It's like we can all like work on things remotely, on the same ideas and like, contribute to ideas. And yeah, if there's like
not only that,
imagine a job board.
Set it up. Yes. And we can set it up to have the derivative content will just flow naturally. So you have to think you'll have the very innovative producers and creators, and they'll have the main cuts of everything. And because everyone's integrated into a full team, then it's like, oh, that's what we're doing. I'm just going to innovate. I'm just going to derivative. I'm just going to make the dance, like we're going to have everything. So Dr. Greg's going to drop the hot anti-vax fucking article of the week. And then we're going to have the animation. We're going to drop an
You went really far. The mic went really far, and now it's disconnected. There you go. You're back now. You're getting closer. Did you join again to the mic or something? Your Bluetooth? I can't hear you.
My headphones are like they're passing away.
There we go. You're good now.
Because I work these long shifts and I use them for 8-12 hours at a time.
Oh, dude, yeah. I hear that.
But yeah, so we'll have the people who generate the real content or the more factual, or there's some people that generate, some people that innovate, some people just do whatever, and we'll have TikTok dance troops to make trends for it all.
There you go. Exactly, man. That's a good idea. I love it. Let's do it. I'm totally down. I think that there's so much opportunity, for real.
Yeah, no, definitely. Definitely. It sucks because I kind of usually come here after Joey's live, but Joey's live ended early today because of a ban.
Oh, really? Oh my gosh, he did a 5-hour stream earlier today, and
that shit was fire.
Yeah. Did you see the Jasmine Crockett's announcement video?
Yeah, I was watching it on there. It really was hard for me to not walk out of work and announce my own run for Senate.
There you go.
I'm not gonna lie to you.
There you go.
I don't know. I feel like even though it would be cool and all the things, I feel like that's probably a very selfish thing for me to try to do, and that I'm getting so much momentum right now with the way that we're doing. I don't know what to call this, I don't know what to call this, because it seems like if I say the wrong word, there probably are some legal ramifications, because low-key this is kind of getting to be a little formal political org, but Shit, that's why I'm hoping for you to come through with Maverick, bro. I need this umbrella to keep me up ready to come out.
Yeah, I think. Speaking of which, I think we're gonna move away. I think MAVA is gonna be the slogan that we use, and then the org is gonna be the thing that I actually registered, which is probably better, safer.
That's
This
whatever
science and freedom
do.
for everyone. But MAVA is obviously the thing, and everyone can say it, and obviously, but the org is safe, So we'll keep it. Well, I don't want to be like, we're a little scared. The admin's scared of getting like a shut down a little earlier than expected. I think that we are going to get sued. We will get sued at some point. But I don't want a cease and desist letter like within the first week. It's like
for MAGA.
Yes, yeah. Maha and MAGA have both been trademarked, so they might come. They would come for us.
Yeah, probably. Isn't that crazy, huh? The president of the United States filing for copyright infringement.
I hope that they do that. You know what I mean? I hope that they do. So that's the whole point. That would be the best PR that we could possibly do. I would represent myself and make content out of the entire thing.
Bro.
And it would be me against the president of the United States in court. It would be unbelievable. It would be so good.
Ooh, not Pamela Joe.
No, it would just be me, baby. Just me. You can't handle the truth.
No, that's funny.
Because
bro.
we can think of a time when we were vaccinated before, so therefore we can make America vaccinated again, but can we ever make America truly healthy again or great again? That isn't alluding to a specific event, while vaccination is. Therefore, make America vaccinated again actually is as a slogan of an actual time and place in American history. I rest my case. Thank you.
Okay, that was closing statement. You know what? I'll tell you what. If I'm on that jury, I might be able to whip something up.
There you
go. You ever did jury duty?
I did. I haven't gotten selected though. Unfortunately, I've tried.
Okay. Yeah, that's crazy. Did you tell them you were a scientist?
Yeah, they don't let us in.
That's crazy, bro. I thought
they don't like it. They don't like it.
The one that I was on, they loved me.
Oh wow, nice. That's amazing. I wonder if it was like, was it a DNA like thing?
No. Honestly, bro, it was it was a family thing. A brother-in-law was working for his wife's brother, and he got fired for allegedly smoking Smeeto. And after the dude fired him, he went outside to fight him. Like the brother attacked the brother-in-law, went outside to fight him, and broke his arm and shattered that shit horribly. So he was suing for damages. And we got my boy $500,000.
Yeah. Bring back thrown hands.
I had to whip it.
Bring back thrown hands instead of bullets. You know what I mean? Bring that back.
Yeah, low key. But man, that shit was crazy, dog. That was one of the most emotional things ever. They was like really mean to the mom and like kicked her out because the mom wanted her the dude to take care of the brother after he did it in the hospital. Was in the hospital. And it was insane, dude.
Yeah, that's gnarly. Hell yeah, man. Well, dude, thanks for coming up. I appreciate you, and thank you for
I gotta go make rounds.
Yeah, thanks for organizing it, and hell yeah, dude. We got our Wednesday at noon is our MAVA or our SAFE Foundation organizational meetings. We do like noon to 2. Like the first hour is kind of like admin and stuff like that, and then the second hour is kind of like a working hour, so we can kind of like work and get things done. So you're always welcome. Just saying you. Yeah.
All right, buddy. Well, yeah, I'm gonna go make some rounds. I appreciate you.
All right. See you, man. Peace. Love you guys. Yeah. Thank you. Yeah, that's cool. That is cool. Thank you, MT. Thank you, man. Appreciate you. Yeah, dude. I think safe is cool. Safe is a really cool acronym, and I think like it works. It works. And MAVA can just be a slogan that we use or put it on merch or something. It'll be cool. But naming the org of that might be a little too much on the front. And I just want to avoid legal ramifications. And we already have everything registered as safe anywhere. Safe is safer. That is the safe pick.
If it plays a role in disease. I'm curious what your thoughts are on that, because it just, to me, it seems like it's quite far away from life. We're not even just in the test tube.
Safe is science and freedom for everyone. Yeah, that's the 501c3 and the 501c4 that we did.
Now we're in the computer. I mean, like if it's going to cause disease, if it plays a role in disease. I'm curious what your thoughts are on that, because it just, to me, it seems like it's quite far away from life. We're not even just in the test tube now. We're in the computer, and we're going off the assumption that this computer represents life. Like these
But dude, I mean, we literally like isolated viruses without computers. Does he realize? He doesn't even realize that. It's like the early 1800s, the tobacco mosaic virus was isolated. That was the whole point. Like he's like decrying test tubes and stuff, but that was literally the evidence that he didn't accept. I mean, we only count, we don't count the 4F. So it's science, science and freedom for everyone. Yeah, exactly. Yeah. You got to do some work, do great. You got to do some work. But it's all good. It could be F with a, oh, that's an interesting idea. I like that. That's a cool one. That's funny.
models represent life, and I guess there's a long history there, but
Sure,
just curious
well,
to hear your thoughts there.
Yeah, the question of whether a virus causes disease or is pathogenic is a separate question from whether the virus exists.
There it is. See how it smiles? See? Oh, this is how he gets his anti-vax stuff in. This is it. Look how smart, look how this smarmy, like, little freaking smile that he has. Did you catch that, guys? Did you catch that?
You have to separate as pathogenic. Causes disease or is pathogenic is a separate question from whether the virus exists. You have to separate these two questions.
Certainly.
You can prove that a virus exists. It doesn't mean that it causes the disease,
Oh my gosh.
suspect it causes.
What an idiot.
And this was acknowledged in the initial papers on SARS-CoV-2, where when they first isolated and sequenced SARS-CoV-2, those initial papers, expressed their opinions that this was the likely culprit. This was likely what was causing people's illnesses, in Wuhan, China. But they said right in there that Koch's postulates haven't been fulfilled yet. They acknowledged that it was yet to be determined. So they had found a key suspect, essentially.
Kung Fu, the guy on the right is a journalist, an independent investigative journalist and researcher. And the guy on the left failed out of biochem major, went back for psychology. So none of these guys have any actual scientific training whatsoever. Maybe they could run the HHS. maybe the guy on the right, either of them could run everything. He's an independent journalist. What's the journalist's name? Jeremy Hammond. Jeremy Hammond.
They found a SARS-like coronavirus, and so that this was, in their opinion, probably not a coincidence. That this was the likely cause of this illness that they were seeing.
So boring,
which was a reasonable conclusion to draw. But Koch's postulates were subsequently fulfilled. There are the challenge studies, hamsters, macaques, different species of animals that they did challenge studies and demonstrated the transmissibility and the genicity of SARS-CoV-2 in those studies. In fact, there's even been a human challenge study. So the first question is, what?
Yeah. I mean, yeah. And sitting in front of a blackboard, a perfectly black, unused blackboard. Like, at least put something up there, dude. At least like put like say like you know what I mean? Like show that you've been working.
Is the cause of when with this is what investigators were asking themselves. There's this strange pneumonia-like illness.
Exactly.
What's causing it?
No, exactly, Kathleen. No chalk dust, nothing, dude. Like, this is not Dr. Blitz, guys. This is, this is a. I mean, it makes it look like he's like doing this out of his middle school. It's just like an empty room. Mrs. Rogers, I'm gonna do the podcast today at recess. Okay? Can I do that? Yeah, I finished my homework. Now I did my multiplication tables. Yeah. Okay. Thank you. Oh my god. Oh my god, dude.
They took patient samples. Typically what they'll do is, you take the patient sample, you filtrate it or centrifuge it to purify it. There is a purification step. And this is another false claim that's widespread out there, is that there's no purification. Well, but there is. They purify the sample. And then they take the supernatant from that purification.
Whoa, supernatant. Yeah, exactly.
And then that's what they inoculate the cell culture with. And then there's another false claim out there that never do.
If you centrifuged it, would the viral particles, wouldn't they be suspended? Would they be centrifuged as well? I guess you could do a diffusion gradient, so then you could capture them. That's what you'd probably do. You wouldn't want the supernatant, because then your host cells would spin at the bottom because they have more mass. Whatever. I'm sorry. Never mind. Sorry. Actual scientist here.
Controls.
I'm sorry.
Scientists never use controls when they do cell culture. Also completely false.
Okay. Yes. Thank you.
They do use controls
all the time.
So they have the inoculated cell culture, and then a control culture that's given the exact same treatment.
Yes.
Only without the inoculation with the supernatant from the patient sample. So they treat the cells. They have to give food for the cells. There's antibiotics to prevent bacterial contamination in the cell culture.
That's gonna be the thing. That's it. That's that he's gonna say that. Oh, antibiotics. So wait. So you're saying that the cells could be damaged from the antibiotics? How do you know that there isn't any cellular damage from the medicine that there was in the culture? Smarty pants.
So they're given the exact same treatment, except they're not exposed to the supernatant containing, potentially containing a virus. And then they watch and they observe the cell cultures side by side, and to watch.
Okay, so no, he said that right. Okay, the supernatant would have the viruses. Yeah. Okay. All right. Okay.
For cytopathic effects, which would be indicative of the presence of a virus in the infected sample, in the affected cell culture, compared to the uninfected control. And so they can compare, you have this cell culture here that's got cytopathic effects, and the uninfected control does not. Well, that's an indication that there was a virus in the patient sample. And then they do further analyses from there to characterize the virus, including you can look at it underneath an electron microscope. You can do whole genome sequencing, etc.
Can I chime in? Because we'll get to the
He's so excited about this. I just it's crazy, guys. We talked about that before. the electron microscopy, there's multiple ways of showing something. You know, in biology, you have to show things multiple ways. You can't just show one. So you need to show like physically that they're there through electron microscopy, and then genetically that is what you think that you see in the space. So, ay.
Electron microscope. I just want to chime in on that. I'm curious about your thoughts on like Stefan Lanka's experiments, in which, in his experiments, claim that he showed that the procedure of using
Stefan Lanka. Like, again, guys, it's so funny. Lanka experiment. What? Oh, Stefan Lanka is a conspiracy theorist and anti-vaccination activist. So we got recommended these terrain theory. These guys don't believe in germ theory, and the guy on the right is an anti-vaxxer, but he also feels like he thinks that viruses are real, but they just don't cause disease. So, oh my gosh, he's an AIDS denial dude as well. He's also he doesn't believe that measles virus is real. Holy shit, dude. Oh, he's also that guy that did the $100,000 euro thing. And then he got beaten out, and then he was required to like pay the appeal. And then he fought it in court for 5 years, whether or not he had to pay that $100,000. That's wild, dude. This guy's
of starving the cells as well as using antibiotics.
Okay, what is his idea? There it is. I knew it.
And that he showed that the procedure of using of starving the cells as well as using antibiotics, or streptomycin, I think is commonly used, actually was able to create cytopathic effects.
What's up? These people are so easy, dude. The grift is so easy. I can just do it. Like, there isn't. I just want to be wrong with these guys one time. I just want to be wrong.
I'm curious about your thoughts on those experiments.
Sure, my first thought is he should publish his research.
Oh,
sure.
Pipette drop. Boom. What's up? Dang. They are so predictable, Brittany. It's exhausting. It is exhausting that they think that they have like these gotchas and that they're actually doing things. Stefan Lanka is this conspiracy theorist that has no idea what he's talking about.
Second thought is he's not explaining why scientists all around the world have done this experiment over and over and over again.
Yes.
And guess what? They do use controls,
wow.
And they can compare uninfected controls that don't have cytopathic effects. And he's claiming, well, no matter what, you're going to get cytopathic effects just from how they treat the cell cultures. It's just demonstrably false.
Guys, like, you just, like, oh my, come on. Like, what would be the experiment to do this? It's so easy. You would have media that you added ingredients to, and you took ingredients out, so you're only changing one variable at a time until you don't get any cytopathic effects. You would measure that by reactive oxygen species, and you could measure that by a spectrophotometer, 96 well plate, super easy, triplicate, biological triplicate, technical triplicate. You could do that in hours. You'd make sure that there'd be an answered question instantly. It was so easy. Like the idea that this is like some groundbreaking idea that he has come up with.
I think one of the big problems from my read of the literature at this point,
My read of the literature. The problem with my read of literature, dude, is the other thing that he actually he talked about, the Rivers review of Koch's postulates, that was from 1937. He's going back to 1937 for current terrain theory, and this guy thinks that he's abreast and erudite on the literature on all of microbiology. That's what this is. That's what's happening right now, guys. Oh, I haven't been doing the live goals. I'm sorry, guys. I just can't, dude. We got so much left. We got 40 minutes left.
Studies
We're finishing it tonight. I don't
on
care what time it is. I don't care. We're finishing it. I might just throw in an adult beverage just to make it even more spicy. Oh, thank you, Island.
When it
I'm
comes
sorry.
to
I talked through it, guys. I want one more time. I have to go back to his thing. God, I like the commitment. It's the dude. I am just I don't even know, guys. I'm not even.
from my read of the literature at this point, a lot of the studies on cytopathic effects, when it comes to describing their use of mock infection, which is what you're
What? Studying the use
on cytopathic effects, when it comes to describing their use of mock infection, which is what you're talking about here as the control. Mock infection would be like similar or same setup in theory. It should be the exact same setup, same.
Bro, this is like he thinks that this is like some crazy thing that they've done. Guys, that's just experimental design. That's not even like that is just proper experimentation. That is nothing to do with what he's talking about. Like for real, man. Like what is he talking about? It's just so embarrassing, man. He didn't. I mean, so what he's saying is that, okay, so if you have in an experiment, you only want one variable at a time. Okay, so like, what if you were injecting something into somebody or exposing something, and in that introduction, you had another variable that you weren't thinking about, that you introduced another virus, another bacteria, your stuff was contaminated, so now there's this other variable that's in there that you don't know about. Thank you, Marleston. Thank you so much. Thank you guys for all the gifts and stuff. Thank you, Island. Thank you for the heart me's. Thank you, everyone. So the idea is that you have it's called a negative control. No template control is another name for it. It's the idea is that it's a negative control, so you don't want the outcome that you're expecting. The outcome that we're expecting right here, that is the positive result, is the virus to grow. So you want a condition that has no virus in it. So therefore, the negative control would be no virus inoculum, right? So you have the media that the virus was diluted in. You have the hosts that the virus are living in, and that's in a separate container. So you have two different things, and then they're both being introduced to two separate sets of animals at the same time. So the idea is like, what if the reagents are contaminated? What if the reagents are causing some type of impact for the animal when you're injecting it? So the idea is that you can show that, oh yeah, it's like the negative control, when it's just the media, when it's just the cells, there is no disease there. But when I add the virus, the only difference between this one and this one is that this one has virus in it. Therefore, the only difference is the virus. Therefore, all the changes can be attributed to the virus. This isn't like some thing that he's talking about. This is just basic like science 101, how you design an experiment, how you actually know what's going on, how you have statistical significance. This isn't like, groundbreaking. This is just this is basic stuff, man. Yeah, exactly. And he's wrong, Rebel. I mean, he's completely wrong. So That's the point, is that you literally have nothing changed, only the virus inoculum. That would be the only variable. So again, he doesn't understand what he's talking about. Also the CPE. I don't know what the CPE. I don't know what CPE is.
Parameters, same cell culture, same whatever, without virus or sample from the individual added. You know, one of the problems is that
Cytopathic effect, but I just went over it. You'd also, okay, so the cytopathic effect, you could easily show that, right? That the cytopathic effect is only by the virus by sequencing, and then also by just measuring the ROS, the reactive oxygen species of the organism that you're going into. So again, it's super simple. It's completely simple. WHO is good. I would say the American Pediatrics Association for Kids vaccination schedules and the American Medical Association are both really good. Yeah, MAGA doesn't like the studies. They also don't like, they don't take this stuff seriously. They don't think that, they don't take politics seriously, so they think that nobody else is taking it seriously. But I assure you, scientists take their job extremely seriously. We make sure that we do the best possible thing every single time. That's why we do it. We try to improve our experiments every single time and make them better. So the idea that we would just like, oh, what? Like, oh, I didn't realize that. Wow, the virus is in both, like the disease is in both things. Wow, I didn't even notice. Like, whoa. It's nonsense, dude. That's just nonsense.
they say, oh, mock infection was used as a control, and they don't actually describe what was done in the mock infection. Now, maybe they think,
Idiot.
you know, maybe the scientists at that point think, you know, we shouldn't have to, you know, if you're reading a paper, you should know what a mock infection is. We did the exact same thing.
It's just negative control.
Personally, you know, when it comes to the literature, I think that is something that should be done. I agree with Stefan Lanka's work, like you mentioned. I think you should publish this research. I agree. I agree with
wow,
that.
Wow.
I believe in transparency, and one of the things I learned about the use of the scientific method in writing scientific papers is that the method section should be extremely, extremely clear, that you can reproduce the results, because reproducibility, of course, is the cornerstone of science, as you've alluded to a couple times here. That is one of my main problems, is that
Yep.
they're not describing the steps in mock infection, and I think that's where a lot of people get hung up. That's a place that I get hung up on, definitely.
Yeah, there's this claim out there that when they refer to mock infected cells, that's not a control. But again, it's just simply false. Mock infected is literally defined as an uninfected control.
It's like so.
It's given the same treatment. The only difference between the infected and uninfected cell cultures is that the uninfected control is not inoculated with the supernatant from the purified patient sample.
He doesn't know what that is.
It's that simple. That's what mock infected literally means. And so
guys, when you're pipetting it, you have like two tubes. You're like, okay, this done, this done. Okay? Like that's literally what it is. You have your control and you're making it at the same time as your experiment. You have two tubes. One is the one is the other. No, they're not scientists, Ruben. The guy on the right is a journalist, but at least he believes that viruses are real, and he's trying to prove it to the guy on the left, which is not a scientist and thinks that viruses are fake, immune systems are fake, that DNA doesn't cause disease. Yeah, Boopty, I got it for one of my. I got it for my videos coming up. I wanted to do like the whole thing, you know, with the mic, you know what I mean? So I got it off of Amazon for $20. It's the worst pipette I've ever seen in my life. What's up, guys? Is this good? You can't really tell that like it's a pipette though, you know what I mean? But it looks kind of cool, doesn't it? Who doesn't believe?
You know, when scientists use the term mock infected, yeah, of course.
Who doesn't believe the viruses exist? How can something spread if it doesn't exist? Wow, Ruben, I love you, bro. I'm not even kidding. Exactly, Ruben. Exactly. Exactly. It's how does it spread if it doesn't exist? Yes. Makes me so happy. Like, you don't understand, Ruben. Doesn't you have no idea, man. It warms my heart that you're here, bro, and you ask questions, and it's like, yeah, thank you so much. It genuinely warms my heart, man. Thank you.
They don't feel the need to define their terms because it's just understood within the scientific community. Everyone knows what a mock-infected culture is. It's the literal definition. So this is, again, it's just kind of a
It's true.
It's one of those claims that are made out there that mock-infected is not a control, but that's literally the definition of mock-infected cultures. It's a control.
It's true, guys. It's true.
Gotcha.
Gotcha. Okay.
What was I gonna ask there? So I'm curious.
It's just a control and the experiment to show that you're not doing harm by doing the experiment. You know, and it's necessary. Everyone does that negative control for everything because otherwise your stuff could just get contaminated by accident. You know what I mean? There's 1,000 different ways that your experiment could. Guys, the only way you understand if you have results is through the controls. Like that is how like that's how you understand what the experiment is through your controls. If you don't run any controls, you have no experiment. That's how it works. That's how we do things. That's why this stuff matters. We actually think about it. Thank you, Ruben. Thanks for saying that, man. I tune in a lot because I've learned a lot. Awesome, man. That's awesome. Thank you. Thank you for being here.
You know, there's a group out there now that it's coming to light, and it's not published yet, but I hope to stay in contact with you, and would love to hear your thoughts on this. They've reproduced Stefan Lanka's experiments, and I think they're going to publish their research, and when it is more out in the open, I'd love to hear your thoughts on it. You know, is that something that is convincing to you at all? Like if there is some sort of step in that process that does produce, you know, through electron microscopy, visible, you know, viral-like particles or exosome, whatever you want to call them, extracellular vesicles visible under like or plaque forming units and cytopathic effects study. Is that something that's convincing to you?
No. Next question. Like, he's talking about spontaneous generation, the idea that an organism could just spontaneously start coming out of nowhere when it wasn't one just because it's sick. And this is God, this is the Pasteur thing where they literally they don't expose the meat and then they expose the meat, and it's obvious that exposing the meat brings the flies that the maggots aren't from the meat itself. Like this is so simple, man. This is so simple. You would just have a piece of meat or something that you just let decay, and you look at it over microscopy over time, and none of the viral like particles would ever come out. You know, it would just never happen because things don't spontaneously generate.
They would have to explain why there's so many published studies.
Because people can still get COVID, Ruben, and long COVID is real, and people can get seriously injured from it. So that's why you just get boosted. It's just easy. It's just an easy thing to do to protect yourself.
Of cell culture experiments in which the uninfected controls didn't have cytopathic effects. I mean, this is their claim. That, oh, well, the cytopathic effects, and this ties right into their false claim that scientists don't use controls. So it's I'm sorry,
Damn.
But it's a hoax.
Oh,
it's a hoax claim.
It's a hoax.
Because again, they're not explaining why scientists all around the globe do these experiments, and they do use controls. And guess
I'm sorry to hear about that, Tiff.
Guess what? They can compare the infected and uninfected controls. The infected cell culture with the uninfected control culture, and there's no cytopathic effects in the uninfected control. And so it's not the case that the treatment given to the uninfected control is what's causing the cytopathic effects. It's observably not true. So they can, you know, they could publish this supposed experiment of theirs. I'd love to see it. If they want to publish it, I'll take a look. I'll look at their methodology. Or first of all, are they doing the same? Are they following the same steps?
I do think there is a problem, and it's a male problem, guys. Let's be real. I think what's happening is there's a confluence of like self-confidence with purposeful ignorance, with male aggressiveness, with being confidently incorrect. And it's just like, it's very rare. Yeah, delusions of grandeur. Yeah, exactly. I think that it's like, because it's easier to just say, yeah, but I know better than everyone else, and that makes them feel more important, and that makes them a more high value man. And you go into red pill spaces a lot, and they talk about high value men. How do you become a high value man? And to them, nowadays, it's not being educated. It's not even accomplishing anything. It's, making people do what you say they're going to do. And I think that it's a power thing where they're like trying to like make people believe something that's wrong, or that they're trying to convince you something that's like so outlandish, and they want to see if you bend the knee. And it's kind of the same way that like Trump, for example, and made a provable false statement in his first Trump won about the greatest crowd size of all time. That right then and there, he knew who was in the game and who wasn't, who was going to lie for him and who wasn't, who actually was going to play ball and who wasn't. And it's important, so a lot of these guys, a loyalty test. Exactly, Bill. Exactly. And that's what this is. These guys are creating loyalty tests for people that are here. And that's why they get so pissed off when they come in here and I push their shit in, right? When it doesn't work with me, and I don't take it, and that's why they get really mad. That's why that guy crashed out like on Friday night. That's why he got so intimidated. Thank you, Alex. So we just, it's the time is now. The time is now to like stop this nonsense, stop coddling these type of people, and just like go, like we're on the offense now, guys. You know what I mean? Science is on the offense, and we're not gonna stop. I don't know how else to say it other than that. Thank you, Marlstone. Thank you guys for all the taters. Thank you.
Steps that scientists all around the world are doing. That's number one. I want to see it. Show me. And then they would have to explain why they're getting a different result from everyone else all around the world. Mm-hmm. So it's just my view of it is that it just like other claims, just like the claim that the CDC admitted SARS-CoV-2 wasn't isolated, just like the claim that scientists never purify patient samples before they do cell cultures, just like the claim that they never use controls when they do cell culture. These are all hoax claims. They're false. Damn,
Hulse. This guy's
these are all the types of claims that are used to support.
Bro, can you imagine being an anti-vaxxer and calling someone else like you believe in hoax stuff? Like what? He's like standing on business right now and just being like, bro, you've been hoaxed too. Isn't that amazing? It's like, bro, you're literally, it's like, whatever. All right. Okay. All right. That's a little weird. Thank you, Marlston. Thank you, guys. Exactly, Ducray. Like, what is this? I love this. This is amazing. And he's actually going hard. He's actually going a lot harder than I was expecting. To actually call him a hoax to his face, like, that is pretty wild, man.
Support this belief in the non-existence of viruses. And so my point of view is this, it's that, there's, if somebody wants to propose some alternative hypothesis for the pathogenesis of what are called infectious diseases, I'm all ears.
Yes.
But it's a situation where, you have to accept the hypothesis that best fits the totality of available evidence.
Oh, totality. Exactly. Now, he actually hit on something. I don't even know if he knows what it is. I don't think that he even knows how right he is. In order to disprove something and actually change a paradigm in science and to actually shift a consensus, you have to show not only why your experiment is correct, but why the totality of all the collected science up until this point is wrong. And you have to encompass that in your new theory. That's why consensus change is so few and far between. That is why it actually happens. But falsification is independent of replacement. No, Rebel, that's not how it works. No, you can't. You don't get to do that. That's not how it works. No, that's not what it is. That is what you have to do. That is the experiment now that is expected, because you have to show why everyone else was wrong up until this point and why your new thing is right. The Rebel, the best example of this is plate tectonics. Right? So plate tectonics up until that point, people had different theories on how volcanoes were made, how islands were formed, and these were all disconnected from one another, right? How ravines were made, all this kind of stuff, how the earth was shaped the way that it is. And then boom, plate tectonics comes, and guess what? It shifts. It's a consensus amongst a ton of different fields, shifts paradigms on every single one of them, has a single cohesive theory that enwraps all of them, and changed everything all in one idea. And it happened in like 1920s, 1930s, bro. It just happened. Like it's like the biggest paradigm shift in science probably ever, and it happened like 100 years ago. So no, you don't get to just be like, oh, okay, if plate tectonics couldn't explain volcanoes, then it doesn't work. That's the whole point. That's fundamentally, plate tectonics has to show why volcanoes were made, why all that stuff exists. Was it falsification or just development in knowledge? Both. You're doing both. You're not just proving something false, but you're actually like contributing to new knowledge. That is the idea about the fundamental mechanism of how the world works. That is what you're doing.
The entire body of relevant scientific literature is consistent with this idea that viruses exist, and some, not all, some can cause diseases in humans.
Some, not all.
And I'm not aware of a single study, not a single one, including all of the studies that are cited by proponents of the no virus theory, the no virus belief, including those papers. There's not a single paper that I've ever encountered that supports this idea. Not one. Not one.
I mean, he's not wrong. He's not wrong. It's like, he's not wrong, actually. It's kind of funny, dude. He's not wrong. It's actually pretty wild.
All of the medical literature supports the hypothesis that viruses exist and they can cause disease in humans. So it's a matter of competing hypotheses, in which you accept the hypothesis that has no evidence, or
no. They're not hypotheses after you have a consensus. This is why I hate using the word hypotheses sometimes. Hypothesis and theory. This is the difference between a hypothesis and a theory. Notice how he's patient to speak, and he doesn't know what he's talking about. So he doesn't have anything to offer. He's just asking questions. So his brain is so open that his mind is so open that his brain's fallen out. He doesn't have anything to contribute to the conversation. He doesn't know anything.
For the hypothesis.
But Rebel, he will still at the end of this not think that viruses are real. So like the due diligence in his position is to ask questions that are clarifying to better understand what they're talking about in order to learn, because the first step in learning is not understanding something, and then that's how you learn. That's the whole point. Being wrong is the first step in learning, and it's okay to be wrong. I love being wrong. I talk about this all the time. If you're not wrong, then you're not learning. So you need to be in a field or you have a career that you love being wrong in, right? That you love learning in. This guy on the left, he thinks that he's 100% right. That's the thing. He's 100% right, and he has closed his mind off to learning so that he is not engaging in this conversation. It's going right over the top of his head. He is not paying attention. He is not learning. He's not engaged. And that's the problem. You learn a lot more from being wrong than being right. Absolutely, May. That is absolutely the first step of learning is being wrong. But the same could be said about Jeremy as well. No, he's obviously, no. He's talking about the research. He doesn't have a pet theory. He's not ideologically entrapped. He's not dogmatic. He's citing the research, which is good. He doesn't know what he's talking about. He should do this for vaccines too, but he apparently thinks that he's an expert on vaccines. I don't know why he thinks that he's an expert on vaccines, but he doesn't know enough about microbiology that he just cites studies. But whatever. Give it to him this time, I guess.
For which literally all of the evidence points to. And so I just have to accept the hypothesis that literally all of the scientific evidence points to.
Understood.
Yeah.
So electron microscopy.
Again, just has no. See, he's not engaged. He has no idea what the hell's going on. He can't even talk about that. He can't even. Can you just say something? Can you ask a clarifying question? Can you, like, why did you think this instead of that? Like, he just isn't there to learn. He's just a stick in the mud right now. Why can't they just make vaccines using aloe or something natural? I mean, Ruben, they do have vaccines out of virus particles. You know, and viruses are natural, so that would literally be what you're talking about. And they do have vaccines like that.
Is another method that's used, of course, to visualize viruses.
Yep.
Maybe you could speak to that a little bit, and then we can dig into that.
Dude, sure. Dude, this is sad. He's getting blown out. He's like blushing here. He has no idea what to say. He's like, I think that I have no idea, and he's embarrassed. He's getting embarrassed right now.
You know, there's that claim too as well that, you know, what scientists, you know, because there's published papers and you've seen it in the media.
Like, I don't even know, like, what would you say? Like, that's such a weird question. Like, scanning electron microscopy. What do you think about that? Like, what? You're just talking about like some random topic? Like, he doesn't even know how to frame this to not get more embarrassed.
Where they show electron microscopy images of the SARS-CoV-2 virus, for example, with its characteristic spike proteins, you know, which give it that this why it's called coronavirus. Corona meaning crown in Spanish or Latin, and you know, so it's got that. It's called a coronavirus literally because of the appearance that the spike proteins give to it of the appearance.
I guarantee you he knows a lot more a year later if you chat with him, but you might get to, bro, I can look at the video titles of his stuff. Like, he's still asking the same basic questions. He has no idea. Like, what are you talking about? Like, we can literally look at his videos, man. Like, he hasn't learned. Epidemics exposed. Polio, AIDS, flu were never caused by germs. 3 months ago. This breaks germ theory. Microbes don't cause disease. They change shape and function. Yeah, it is. I think it's this guy or I actually don't know. Jamie Andrews might be the $40 a month Substack guy. I don't know. These grifters make so much money, dude. It makes me sick. Makes me sick to my stomach. What are his sources? Ruben, my sweet summer child. He doesn't have them. We're going to go through all these guys' videos, guys. We found our lol cow for a little bit. For the next week, we're going to go through this guy's stuff. And so we'll see. We'll see. We're gonna answer all these questions. This is his best case scenario. Okay, this is the best he has because this is a guy that actually knows enough to know that he doesn't know about it, and because this is important, Heather.
Appearance of a crown. So this crown-like appearance is why it's literally called a coronavirus. And so we've all seen those images, I think. And there's a claim that, well, that's not really a virus that scientists are...
Ruben, I love you, dude. Exactly. Who makes an entire YouTube channel on something you're not an expert in? Exactly, dude. Exactly, Ruben. Exactly, dude. I can't, like, exactly, brother. He's a psychologist. The guy on the left went to school for psychology. He didn't even make his YouTube channel about psychology, bro. He actually has a master's in psychology, doesn't make his YouTube channel about psychology. And like come on. Like, isn't that wild?
Photographing. It's exosomes. Well, this gets into additional claims that are demonstrably false. So, for example, the claim that, well, scientists can't...
And Hazel, you've done it. Exactly. The grift isn't in psychology. The grift is in this. The money is in this. He makes money doing this, so that's what it is. And that's why these guys like get entrapped in here. Like, they are fervently connected to what they find, like to their ideology. They're dogmatically trapped, right? They can't change and be like, oh wow, microbes are real. I'm totally wrong. Pathogens are real. Oh my gosh. If it found out some way that like there are some vaccines that did cause a bunch of harm, I'm not going to advocate for them, right? Even though the good example is the Johnson & Johnson Johannes vaccine, which didn't even harm that many people, by the way, but still VAERS and VSD had it, so they pulled it. Like it still was fine for a lot of people, but that did more harm than others. If there was some other technology that came out, like gene therapy technologies, that was T cell editing, and you don't even need to have vaccines anymore. Do you think I would like talk about vaccines? I'd be like, no, we got this other technology. This is great. We don't have to worry about that anymore. Remember the time that we got shots in the arm all the time? That was crazy. Now we just get one shot for our entire life and we're done. Isn't that amazing? Exactly, K-Bug. Yeah, there's a ton of people in here that actually got the Johnson & Johnson and they're completely fine. So I don't even want to give that as a real example. But my point is that like if it did happen, I'm not married to that position, right? So it's like these guys are, though. Their entire personality is being anti-germ theory and like terrain theory and stuff. So they literally cannot move their position.
Can't distinguish between exosomes and viruses. And you can go and look up that paper that's cited to support that claim, and you can see that the entire thrust of that paper was that actually it's true that if you just are using electron microscopy, they're difficult to distinguish. Granted. But the whole thrust of the paper is how scientists can and do and should distinguish between exosomes and viruses. So there's additional analyses that scientists do to be able to distinguish, just looking.
Yes, Ghostbusters. One of the guys that is the biggest anti-vaxxer, for example, John Campbell. Okay. This guy, John Campbell, you guys might have seen him. The guy is a notorious goes as a doctor, says that he's a doctor.
You're most welcome to this.
Okay, this guy right here. This guy. Probably saw him. This guy, a retired nurse, has a PhD in digital marketing, calls himself Dr. John Campbell, and gives medical advice, disproving everything about the COVID vaccine, all, microbiology, all that kind of stuff. The grift is real. The dude has 3 million subscribers on YouTube, bro. 3 million subscribers on YouTube. Like, it's just, bro, like, the grift is real, ladies and gentlemen. The grift is real.
At the electron microscope, images, an exosome could look very similar to a virus. Absolutely. And so this is the point that was made in the paper, but it's completely that, statements like that from the paper are taken completely out of their context. And if you just go and read the whole paper, the whole point of the paper is about how scientists can and should differentiate between the two. So the claim that the claim being made to the claim that this paper is being cited to support is that scientists can't distinguish between the two. And you go and read the paper and it talks all about how scientists can distinguish between the two and how they should do that. So this is an example of kind of like these hoax claims that are out there. And again, claims that are actually contradicted by the sources that are cited to support them. Yeah.
Gotcha. Yeah. And I appreciate that honest take. I would agree that with what you're saying there about electron microscopes. It's not enough. It's a piece of the puzzle.
Precisely.
For virology's claims.
Yeah. Again, that's a really important point. It's not just about isolation or just about electron microscopy or just about whole genome sequencing. There's a variety of different tools and analyses that scientists do to be able to identify and characterize viruses.
Mm-hmm. Yeah. Definitely. And the last big one.
Ruben's on fire tonight, dude. Why talk to a journalist instead of a scientist? That's a great question. Well, Ruben, he's going to get a chance to talk to me real soon. So there you go. We'll see how he does. It's probably not going to go very good to him. I doubt he accepts.
One is immunology, serology, antibody research. That's sort of the last big one used to identify antigens. Things like SDS page, electrophoresis, stuff.
What?
Different. There are many different types of ways to do that. Western blot is a big one. There's a ton of different types of blots too. Anyways.
Okay.
That's another big one, and that's.
I'm going to contact him for sure. I'm going to email him. Yeah.
One of the things, it's all very reliant on each other to sort of prove each other correct.
The journalist is doing okay, honestly. Yeah, he's saying SDS page, which is, yeah. I mean, he's just rambling. He really just is Mad Libsing vocab words he knows. He doesn't understand why or how. He's talking about like, oh, there's lots of different directions for the north. Yeah, there's a north, south, and west. Blot. So it's just. Southern blot's DNA, northern blot's RNA, western blot's protein.
Correct. That's like you were pointing out there. With antibodies, one of the big things, and this may be out of our scope here. One of the main claims. Curious if you've heard about this. Have any thoughts? Antibodies displaying some sort of non-specific behavior, and so shouldn't be used again as a
nice
gold standard to detect whether or not there is some sort of virus or viral material present. I'm not sure if you've encountered that claim whatsoever, but
so non-specific in terms of that an antibody might bind with so in other words, the assumption might be, okay, well, this antibody bound with this particle, therefore that particle is such and such virus. when actually that antibody might also bind with other particles. Is that what you mean? Is that the thrust of it? Okay. Actually, I don't know that I've heard that. But that's a legitimate point that, yeah, sure, not antibodies might be not necessarily specific. There might be non-specific, but in general, antibodies do have specificity to whatever they are developed in response to. So, for example, when you first experience an infection with SARS-CoV-2.
I know exactly, Foopty. I know, like, it doesn't make any sense, dude. Like,
and I'm just picking that because it's kind of at the
they're
forefront
gonna have
of my
binding
mind.
affinities to other motifs, and that's okay. That you're still not gonna be able to, like. It's like more than one antibody.
When I talk about this issue, but pick
more than one binding receptor on the cell.
Virus, but I'll speak on that also because I've researched it so extensively.
Sure.
Because one of the reasons I spent a lot of time researching the immunology on SARS-CoV-2 was because of the claims that were, throughout all of 2020, in the summer of 2020 through the fall, right up into the point when they.
Oh, dude, does he feel bad for him? And now he's gonna go anti-vax on him, and he's gonna try to rescue him because he feels bad because this kid just got walloped.
The vaccines received emergency use authorization.
Damn it! I just like another second. I didn't even need to pause it to be. Oh, here it is.
We were bombarded with all these headlines claiming that people were losing their immunity to COVID-19. And I was writing to debunk all those propaganda claims that were, of course, designed to create fear and to manufacture consent for the experimental vaccines that were under.
Chomsky is just like rolling over in his grave right now, just like, oh my God, why are you saying that? What are you talking about? Manufacturing consent? You just, what? Are you out of your mind?
Production. But anyhow, so I'm just picking this particular virus because it's in front of my mind when we talk about this issue, and also because I know so much about the immunology of it. But pick your virus, but I'm just going to pick that one. So, so when you're infected for the first time with SARS-CoV-2, although there is some level of cross reaction with prior infections from common cold coronaviruses. So your body does have some, because you've experienced coronavirus infection with, common human coronaviruses that cause the common cold. You've got some immunity to those viruses, and there is some like cross reaction in terms of your immune response from that past experience. Not necessarily terribly protective against SARS-CoV-2 because it is quite different from the common cold coronaviruses.
It's a novel virus.
So it can take a couple of weeks for your body to actually develop antibodies.
There are coronaviruses that go around, that circulate, yeah.
That infection. It can take several weeks. I mean,
SARS-CoV-2 was a novel coronavirus, though. So it was like, it's a novel one. So that means that we had no antibodies for it. So he's wrong on that. But there are coronaviruses that are in circulation. But SARS-CoV-2 was a novel one. So we didn't have any. That was the whole point.
So the body starts going through this process of producing antibodies. And, it might start out producing antibodies that really don't have high affinity to the virus. But in time, the antibodies that are
novel because it's new. Novel meaning new and not seen before. That's why the term novel is used.
Effective in
versus like one that's like a couple mutations off or something like that our body would have already known. Yeah, Floopty. Yeah, exactly.
Binding to in neutral.
exosomes. Looking at it. Exosome looks like this, guys. So it's like a heat shock protein in there, a protein that's like what, 30 to 150 nanometers. That's a larger virus. It's a fairly large virus. But the idea that we would just see that and be like, that's a virus, and then that's it, and then you never do any other investigations. You just that's it. Like, I mean, come on, that's just nonsense.
Neutralizing the virus are selected for, and so the body learns and trains itself. The immune system trains itself in how to produce an effective response, and so. And then what happens is, once you have essentially cleared the infection with SARS-CoV-2, also what happens is you develop, which is typical for infectious diseases, is you develop a long-term immune memory. So you have long-term bone marrow plasma cells. Mm-hmm. And this is one of the claims exactly that they were making in the media and the headlines where people were losing their immunity, and that was based on this observation that, well, people had high level of antibodies after the acute phase of infection, and then it just started to plummet, and so people were losing their antibodies, and so therefore they were losing their immunity. And this was a bunch of ridiculous nonsense that just was like contrary to like immunology 101. And again, I'm no expert, but it's just like basic immunology.
It does happen. We do.
That's completely normal for that to happen after.
No, he doesn't, Rebel. He literally was just talking about it again. Like bro, like these are microscopy photos that I had. Like, I've used microscopes before. You don't know exactly what it is, but the idea that there would be some foreign body that was in there that you just don't even investigate and you just assume that it's a virus. I mean, that's nonsense, dude.
For the acute phase of the infection, it happens with every infection, where you don't maintain that high level of antibodies after the acute. Mm-hmm. Yes, there's always going to be a decrease in the antibody levels, but every study that looked at it after that found a continuation of detectable antibodies in people's blood who had recovered from COVID-19. And scientists also confirmed that, yes, that infection induces long-term bone marrow plasma cells. So you have long-term immunological memory, so that if you ever encounter that virus again, your immune system doesn't need to learn all over again how to fight it off. It already knows, and so those cells are able to rapidly produce antibodies again that are high affinity to that infecting pathogen. And this is where, we talked about variants earlier. This is where, another issue with the variants is that, so people's immune memory would start producing antibodies that are specific to whatever, variant that they were infected with. Let's say they were infected with the original Wuhan strain. And later they were infected with Omicron. So their body was able to rapidly produce antibodies, but they weren't as specific to the Omicron variant because they were developed in response to the original Wuhan variant, right? Mm-hmm. And so, but here's another really important thing is that with natural immunity.
Well, it's a good thing he doesn't have a big following. Ruben, you are on fire tonight, bro. Oh my God, this is amazing. It's so good, dude. Love you, Ruben. That's so funny, dude.
Community, the body is able to update itself, and so people who are maybe had a reinfection with Omicron, their immune systems were able to rapidly update that antibody response to be specific, to have greater specificity for the spike protein of the Omicron variant. Uh-huh. And this is something that people who received the vaccine weren't able to do, because with the vaccine, there's a problem that's known as original antigenic sin, where the initial priming of...
Bro, are you serious right now? I mean, I just want to break this up. Electron microscopy, SARS-CoV-2, a challenging task, Goldsmith, 2020. This is it. This is gonna be it. Okay. let's do this. Watch. Okay, so we have the paper, all right? Cited by 28. All right. Okay. All right. So this is a comment. All right. So it's not a research article. In May of 2020. That's 6 months after the outbreak. Okay. Cited by. You go to the cited by section. Boom. And then you can see these are reviews. Enhancing the stability of bacteriophages using physical, chemical, and nano-based approaches. Why misinterpretation of electron micrographs in SARS-CoV-2 infected tissues goes viral. So they're actually even talking about that. Ultrastructural examination of lung cryoblasts from a series of fatal COVID-19 cases hardly revealed infected cells. Molecular signaling pathways, pathophysiological features in various organs, and treatment in SARS-CoV-2 infections. So none of them are saying like, wow, that paper was amazing. That the next thing was like, we couldn't figure it out and we have no idea what's going on. And bro, are you fucking kidding me? It's a figure with a bright field image. This is like your gotcha. This is the We read with interest the correspondence of Zarga and colleagues on the possible infections of endothelial cells of SARS-CoV-2 using EM images as evidence. However, we believe that EM images in the correspondence do not show coronavirus particles, but instead show cross-sections of the rough endoplasmic reticulum. These spherical structures are rounded dark dots, which might have interpreted as spikes on coronavirus particles, but are instead ribosomes. The purported particles are free within the cytoplasm, whereas with a coronavirus infected cell, accumulations of virus particles would be found in membrane-bound areas in the cisterine of the RER Golgi area, where the spikes would be located on the inside of the cistral space. In addition, cross-sections through viral nucleocapsids are not seen in the interior of the structures as would be found with coronavirus particles. And this is bro, in this same thing, it says viral isolate grown in cell culture. So even your own thing is saying that they literally... No, it's not published. God. It's figure. Viral isolate grown... This is... They're showing here. This is what it would actually look like. They're trying to correct another person being like, hey, when you do this an experiment in the future, make sure you're looking for these things like this here. This is a viral isolate grown in cell culture. So they're trying to help it, and they're trying to say this is what it would actually look like. So you're not... Your own evidence is showing exactly what it is. Yeah, exactly. Yeah, they could misinterpret it, but that's why they're posting that, and it's showing here that they have and they can. So it's like you can do it, and that's it. They're doing it right there. So, you know what I mean? And these are all showing yeah, I mean these are all talking about like the doing that. this is just a correspondence, dude. This is just this is like a letter to the editor. This isn't a publication. Okay. This is just a letter to the editor, bro. This isn't like oh my God, this is impossible. Like, this is a defeat for science. They're just like, hey, by the way, this is make sure this.
Of the immune system can prejudice the immune response to any subsequent exposure to whatever pathogen. And so people whose immune system were primed by infection have a robust, long-term adaptive immune response, whereas people whose immune systems were primed by vaccination don't. We've seen, of course, there's the rapidly waning immunity with the COVID-19 vaccines, and then there's this problem of original antigenic sin, where they notice that if they did experience an infection, what they call a breakthrough infection with, say, the Omicron variant, that the antibodies generated don't adapt. They stay fixated on producing an immune response to the extinct spike protein of the extinct Wuhan strain, at the opportunity cost of not producing a more specific response to the newly infecting variant. And so that's known in the literature as original antigenic sin, or
Thank you, Jenny.
Sometimes it's called immune priming, which I think is a really stupid name for it, because
Thank you,
every
Catherine.
immune reaction to any new pathogen is immune priming. So it matters, like, what is the priming? What is, is it priming from infection, so natural immunity, or is it priming from vaccination, which is different? And so just immune priming is kind of a very nondescript label for this phenomenon, but it's also called original antigenic sin, which I think is more fitting and a better description for it.
Cool.
But anyhow, yeah, I think so this kind of speaks to the we were talking about variants earlier, so that kind of ties together in terms of like antibody responses, and there is a question of specificity of antibodies to a pathogen. But the whole aim in terms of the immune system's function is to produce antibodies that are specific, and of course they're not always, and this can be a problem. And it's why people can get reinfected with variants even though they've had an infection before.
Non-specificity of antibodies doesn't necessarily contradict that either, though, because like if we have they talk about the big five, that's what we learned about in my introductory microbiology classes. It doesn't necessarily go against the theory at large of having some sort of
I might have been his professor, guys. You know what I mean? That could have been me. Could have been me if he was at UCR. Dude, if any of my students actually became anti-vaxxers, I would be, or didn't, I never even thought at that moment in 2022, like I did the germ theory stuff, like in terrain theory, I wasn't even aware of. Anti-vax in general, yeah, but to like just deny all of microbiology, I would have been shocked. It's okay, Rebel. I mean, it's wild, man. It's wild.
Immune response either, which I think is interesting. Used as a tool, it is a source of irreproducibility in the literature, which I think is problematic. And there are a lot of companies out there that are trying to remediate that. Like it's a huge problem in science, is the non-specificity of antibodies, because it's used across the board, in science and scientific research. So something to look into. I'm really curious about.
I don't know what you're thinking of, but I'm thinking of, like, for example.
Yeah, what was that brain belch? Like, what was that? That's a great question, Ruben. How can people not believe in the immune system? What else would we have? Yeah, exactly. So the person that I talked to that was talking about this, basically they thought that we just have like he called it the lymphatic system. And that was it. We didn't have any like T cells, B cells, antibodies, all that kind of stuff. It was just a series of stuff that's cleared through your body in the lymphatic system. I have no idea, dude. It's actually wild.
Example, maybe this is completely different from what you're thinking, but like for example, they're trying to develop vaccines for Lyme disease, and the FDA is using antibody tests to kind of determine, whether those vaccines are effective. But the problem is that, a lot of people who are infected with Lyme don't develop antibodies. And so if you're just looking for antibodies to Lyme and it's negative, it doesn't mean you don't have Lyme disease, for example. If you look at vaccine licensure, there's a problem with looking with the use of antibodies. Because the whole purpose of the vaccine is basically to cause the body to generate antibodies, but of course antibodies are not the immune system's only response to pathogens. There's also cellular immune responses and T cells, and SARS-CoV-2 is actually a very good example of how the cellular immune response is actually more important than the antibody response, and this is something that the public health establishment only seemed to discover after they had to finally admit, after it was just absolutely clear in the data that the vaccine effectiveness against infection waned rapidly. And suddenly,
Dr. Cohen has his own shop. Wow, you can eat right and have your own stuff with your own things. That's incredible. Wow. All my things. Wow. How convenient. Nobody else can keep you healthy but me. I have the knowledge that nobody else has. Wow. This isn't suspicious at all. I know, dude. Look at this. Look at, he's just selling whatever it is. Look at this. Like, guys, like, there's no, you can't actually, nobody actually takes this serious. DMSO. Look at $109. What? Marine plasma drinkable seawater, $55. Are you, there's no way. Se 750 mils. It's not even a liter of seawater for $55. Are you out of your mind? The maximum recommended dose per person is 50 mils. Ooh, a subscribe and save. You're right. Fuck me, Mackenzie. You're right. You're fucking right. Oh no. It's sold. Oh, guys, I'm done. I'm done. This world, I don't even know, man. Bro, how can you be so hyper accountable to science? And then this guy who's like selling something, he has every financial incentive for you to think that science and medicine and nature isn't giving you any type of, you know, reality except for him. Like how can anyone not? It's so obvious. Like it's so obvious. How can people seriously think this is legit?
Suddenly they discovered cellular immunity and they were like, oh, but don't worry, you might get an infection.
324 million, dude. Oh my God. Yeah. I'm like sick to my stomach, dude. I'm not even kidding. Like what?
But you still have.
Imagine being like an idiot and actually dedicating your life to science and actually trying to improve things and actually trying to fix problems. Like imagine doing that. Like what an idiot. Dude, Rebel, holy fuck, dude. Think of the profit incentive and the profit margins that they're making. Imagine like the physician and your naturopathic doctor and imagine if you went to the doctors, your PCP, and your doctor was a Pfizer doctor and he sold you only Pfizer things. And you go in there and he's like, this is what you need to take. This is what you need to have. Like, you wouldn't be suspicious of that if he was only prescribing you Pfizer things, Pfizer medicine. You wouldn't be suspicious of that. And then someone's like, oh, you don't have to buy the Pfizer medication. But the whole point is, he's saying that this is the only thing that keeps you safe. This is the only thing that keeps you healthy. This is the only thing that keeps you like, okay. That's the whole point. He doesn't recommend anything else. God, man, how can people not see this? It's so obvious. Like, you gotta like
protection against the disease, and your symptoms won't be as bad because of you have T cells. And suddenly they discovered T cells, right? But of course, you know, this is something that they had always known about, and they just never educated the public that it's not about antibodies. It's not all about antibodies are important, you know, in terms of preventing a circulating virus.
Bro, it's not this. It's not, whatever. It's people get hurt by this. This is not FDA approved. This is not FDA regulated. You don't even know what it is that you're getting. A lot of times, these not FDA approval stuff, guess what, bro? They're just like 90% some random stuff, and people overtox on them, and they go in the hospital, and they do get poisoned from it.
From being able to bind with the cell receptors and enter and penetrate the cell membrane to gain access into the cell, and then using the cell's mechanisms to replicate itself. Because you know, the virus is an intracellular parasite. It requires host cells to replicate. Allegedly. And again, that's why scientists use cell cultures to isolate viruses. But you know, but the antibodies are just one factor in you know, a broad range of immune responses.
No, some are, bro. Yes, they do regulate some of the stuff. Yes, they do.
Responses that are important. So so I'm not sure if that's along the lines of what you were thinking, but we might have some agreement in terms of, you know, if you're just looking at antibodies, it can be very deceptive, you know, or when they say that, well, if you have a certain level of antibodies, that means you're protected. Well, that's not necessarily true. You can, in fact, with SARS-CoV-2, study after study was showing that, you know, that patients who had severe disease was correlated with a high level of antibodies, whereas patients who had very mild symptoms or no symptoms at all, they had an infection but no disease. And that's another important distinction. You can have an infection without disease. And so they were finding that patients who had, who their immune systems just absolutely
Bare necessity. That's a really good question. Do they make this kind of money in the countries that have universal healthcare? That's a good question. I don't know. That's a really good question. Yeah.
handled the virus without hardly any problems whatsoever at very low levels of antibodies. And this is explained by the importance of the cellular immune responses. And so there was actually the contrary correlate. They want us to think of you need a high level of antibodies to be protected against this disease.
No, bro, he's saying that the Morgan Stanley was the one that did the wealth management stuff, not that he's working at Morgan Stanley.
In fact, in terms of natural infection, the opposite was actually the correlation observed, were that people who had high levels of antibodies were deficient in cellular immunity, and so they developed more and more antibodies, but those were also the patients that had the most severe disease. And so absolutely, if you're just looking at antibodies in this context, there's a lot of misunderstanding and misconceptions about that. But I don't know if that has anything to do with the line of thought.
Well, that's an interesting observation at the very least. I think that's something that merits a little more digging into for sure. Because that's a really interesting observation, to say the least, for sure. And it sort of circles into one of the last things I want to ask you about. I got two more things if we got time, but the one thing I want to ask you about is the PCR test.
Oh God!
Glad you brought it up.
Yeah, I think that's an important part of our discussion for sure. So maybe I'll let you have the floor, and then I can chime in.
Well, sure. I mean, as soon as you mentioned that, my thought is there's a lot of confusion out there. I've had so many of my own readers. It's
probably
not so much anymore, but in 2020 and 2021, throughout that period of time,
is PCR real?
Just so many of my readers were so confused about kind of the distinction because they were seeing how the PCR tests, the tests that were being used to diagnose COVID-19, there was systematic fraud in the way they were utilized to diagnose COVID-19. Because
here's the
PCR, those types of PCR tests just detect viral fragments, so they can detect RNA from the virus, but they don't look at the whole genome sequence. So they can detect just fragments of viral RNA, and you can have fragments of viral RNA in your body, and it doesn't mean you have an infection. It could very well mean that maybe you were infected before, and your immune system has already fought off the virus, and you don't have an infection at all, and you're completely symptomless. There's no infection. You're not contagious. You're not a threat to anyone. And they were calling people, who had just like fragments of RNA in their system. They were calling them COVID-19 patients. And well, that's false.
No, I mean, they have SARS-CoV-2 positive. Doesn't mean that they have COVID-19. So again, SARS-CoV-2 virus, COVID-19 is the disease caused by the virus. Distinctly different things. God.
So, and to utilize these tests in that way.
Honestly, Ruben, I have no idea what he's talking about right now.
Was really systematic scientific fraud in the way they and the other thing is you could even have an infection, but that's not necessarily the cause of the disease.
Sure.
Which I'm sure you agree with, that somebody might have the PCR test might be positive for viral RNA from SARS-CoV-2. It doesn't mean that that's the cause of the disease. Again, there might not even be an active infection in that case. And so to describe anyone with a positive PCR,
I know he was just mopping the floor with that guy, and then Puff, what's up, dude? He was just mopping the floor with them, and then now he just feels bad. Now he's going to a place where they can both agree. Like, it was just kind of like compassionate surrender.
PCR test as a COVID-19 case also overlooks the distinction between infection and disease, because again, you can have an infection and not have the disease. The disease is the symptoms. That's what a disease is. The clinical disease is the syndrome, the symptoms that you get from having an infection. If you don't have any symptoms, you don't have the disease. So the disease doesn't actually develop, and that's another important distinction. So the way that they were counting COVID-19 cases was really premised on this scientifically fraudulent use of these PCR tests. And so a lot of my readers and
No, it's the opposite, Ruben. We actually, the WHO and the China's primer set for the PCR tests at the beginning, those are actually fine. It's because Trump was so stubborn. He decided that we should have our own ones that weren't tested and had a problem with them. And that was the problem that was originally associated with it. So it was the China's and the WHO primer set was perfect.
a lot of people that I saw, it's on social media and whatever, engaged with, were just like, they were seeing that, and then they somehow like took that over to the use of genetic sequencing, genome sequencing, which uses PCR technology. And so they were kind of saying, well, so the PCR tests are being used in a fraudulent manner, and therefore, the whole genome sequencing uses PCR technology, that must be fraudulent too. And so people were kind of making this leap of logic in because they were hearing that type of thing from others who were kind of propagating these types of claims. And so I kept finding myself having to kind of clarify, hey, look, yes, I'm saying that whole genome sequencing proves that this virus exists. It doesn't mean I'm saying that these PCR tests are being used in a legitimate fashion to diagnose COVID-19. It's like people would would make that assumption and attribute that kind of argument to me. And I'm like, no, that's not what I'm saying at all. So anyway, when you bring it up, and I say I'm glad you brought it up, that this is kind of what's going through my mind because of that experience of mine, dealing with lots of people on social media, or even my own readers emailing me and kind of either just confused about it and asking me, or challenging me and confronting me about that. And so it's really important to understand the difference between those PCR assays that were used for improperly for diagnosis, and just the PCR technology itself, which is legitimate technology. And so people need to understand that distinction.
And I'm glad we agree on that, and I think I'm curious if you looked into this at all, but they were running the tests at a much higher cycle threshold than is scientifically acceptable.
Well, that's the other factor in that, is that they were running the PCR test at such a high cycle threshold. So for viewers who aren't aware, so the way the PCR tests work is actually, so it's reverse transcription polymerase chain reaction, so RT-PCR. And so they because the virus is an RNA virus, and so they have to reverse transcribe.
It's reverse transcribe RT-qPCR. It's quantitative. Never mind. Whatever.
RNA into DNA, and then they use cyclical amplification of the DNA up to a what they would consider a threshold of positivity. But they were running the test at such high cycle thresholds that they were turning up positive for just tiny fragments, tiny amounts of viral RNA that did not represent whole viable virus. And it's such high cycle thresholds that it was known that didn't represent.
Yeah, it's not true. I mean, again, you can have you do a serial dilution so that you can know. Again, this is just nonsense. I mean, quantitative PCR just means that you have a probe in there, so you're actually knowing what the amplification cycle is. So that's the whole idea. Yeah, this guy has no idea what he's talking about.
That in all probability, they were running the test at such high cycle thresholds that they knew that it probably did not represent somebody who had an infection in somebody.
Yeah, it's just not true. I mean, again, if he's saying it's non-specifically binding at those thresholds, then I mean, it's very easy. Every single one, it has a melt curve on top of it. You run a melt curve, and it basically checks and makes sure that the length of the amplicon is exactly what you're expecting, because if it's non-specifically binding, the amplicon is going to be varying lengths, and it's not going to be the exact one that you want. So it's such an easy QC. As if we wouldn't know that. You can have very specific, usually 40 is the highest you would ever run it. You can have very specific 30 degree 30 CT, 35 CT. That's completely fine. It's completely fine. Yeah, the wastewater data, exactly.
Who was contagious, and they knew it.
Absolutely.
So,
Absolutely.
and this again, I'm glad you brought up the cycle thresholds, because again, it just speaks to how widespread this scientific fraud was.
Yeah, what an idiot. Not a fraud. It's not a fraud, and we weren't widespread. God, dude. Yeah, she's just crazy, man.
In terms of diagnosis.
But he's an anti-vaxxer, so of course he thinks that a widespread, worldwide, global science conspiracy is definitely possible. That's that in this guy's wheelhouse. It's there, and now it's coming out. Especially now that all the anti-vax stuff is coming out. Now he's all into it, and he's into his safe space.
What they called COVID-19 cases, and then of course anyone who was a COVID-19 case who died, they considered a COVID-19 death.
Yeah.
So the amount of scientific fraud in
we got
that
10
regard
minutes.
Was
again, holy shit. Can you be just like, not hard, dude? There's more expected deaths than we had before. Really not that hard.
Absolute. But again, a lot of people were confused about that when it came to the PCR technology being used with whole genome sequencing, and just kind of not understanding the difference. But yeah, I mean, I'm absolutely in agreement about the systematic fraud involved with PCR tests.
Yeah.
There was none. Again,
okay,
nonsense, dude. Nonsense. Holy shit.
If I can ask you about one more thing. I'm curious about your thoughts on like real world contagion studies. And the big one for me, and the first one that I ever read that was the most convincing for me on sort of the train versus germ argument, was the Gallup's Island study that was done back in by Rosno back in 1919 for the Spanish flu. And, he took a bunch of, I'm not sure if you're familiar with the.
My recollection was 1918, but I think pretty sure we're talking about the same study. Yeah.
I think we're talking about the same study. 1918 is correct. Yes, you're right. But yeah, I'm curious. So I'd love to hear your thoughts on that. The listeners are quite familiar. It's the controlled contagion studies, essentially, of.
Yeah, that's the one I've most often seen cited.
Fucking hilarious, dude.
But I don't recall. It's been some time. I think I've looked at that whole study. Pretty sure I've managed to get a hold of it and look at it, but I don't specifically recall.
You didn't look back in history at all, dude. You didn't look back at all, dude. You can't even. The paper's not even available. Get full text access. You bought it? You looked back in history? Send me the PDF. You didn't do anything. You didn't look back into anything.
Other than that,
you don't have it bookmarked. It's you paid for it. Okay, sure.
The important point to make is that, in 1918, they knew practically nothing about viruses, and they knew practically nothing about viral transmission and contagion. And they had some sense that there were these tiny particles that were, transmissible and could infect people. But their knowledge of virology and immunology was just primitive and basic. And we've had, over a century of scientific research since then, with many.
This is what you're thinking. This is what you're thinking to disprove all of microbiology. This stuff. You're going to this, bro. You think this is good data in order to disprove everything. This is what you're going to. I just want to make sure. Bro. Like, okay. like, so what do you think has more complete methodology, materials and methods sections, this or modern papers? Like, come on, bro. This is a joke.
Many studies demonstrating transmissibility demonstrating existence of, as well as contagiousness of viruses, including, as I mentioned earlier, with SARS-CoV-2, there's that claim that Koch's postulates as understood today. Of course, Koch's original postulates were just plain wrong. So, and then you mentioned Rivers' kind of, modification of Koch's postulates, and Koch's postulates have been kind of modified over time to become, more specific and more accurate and more valid in terms of how do you determine whether, particular agent causes a disease. So when I refer to Koch's postulates being fulfilled in terms of SARS-CoV-2, I'm talking about, as scientists understand Koch's postulates today and the modern research methods and modern understanding of the steps involved to actually demonstrate causality. So there were animal challenge studies, and even as I mentioned, there was actually one human challenge study with SARS-CoV-2 where they deliberately infected human subjects and then observed the pathogenesis of disease in those patients. So yeah, I mean, this claim that people, I see people citing that 1918 study as though that study proves that there's that the flu wasn't contagious, but it doesn't prove that. I mean,
Gotcha.
I guess that's my main point is that study doesn't prove that.
Sure, got it. And that's good, and I want to tell you something about my experience in university, because I just graduated from my degree 2022. So when I was in microbiology, when I took all my microbiology classes, they taught Koch's postulates. And I can appreciate what you're saying. Like I can. You know, that's an area of distrust for me personally.
Okay.
And I think that there's this it's not right in my mind that university students are going in, they're learning about Koch's postulates as being the gold standard way that things are approached, when now in scientific fields something different is being done. I think
that's
a huge
not.
problem.
They taught you like Koch's original postulates?
Original postulates.
As though that's still valid today?
What we were taught in multiple classes, not just one micro and high level up class, 3,000, 4,000 level courses. They were teaching Koch's postulates.
Oh my gosh.
So just as an example of what I mean when like that his original postulates are invalid and now known to be incorrect is, for example, he had made an assumption that if there's an infection, therefore there will be with a pathogen, therefore there will be a disease. And so
we know that's not true.
You will not find, in other words, you will not find that pathogen in anyone who doesn't present with the disease, which is completely false and was subsequently disproven, which is why one of the reasons I think Rivers was correcting the original Koch's postulates and updated them with the improved knowledge of scientific thinking. So I'm really shocked to hear that they're actually teaching the original Koch's postulate as though they hadn't been like refined over over a century of scientific thought and knowledge, that hadn't been refined. That
he went for a biochem undergrad and then flunked out and then went back for psychology.
That would also, if I was you going through that experience, would also create distrust with me, because that's crazy. Yeah, that's crazy. But I guess it shouldn't surprise me that they would teach something like that in school.
So the other amazing thing was, like, I was taking genetics too. My degree was largely genetics. I took biochemistry and molecular biology, so it was mostly genetics. I was in a genetics course during the real height of COVID, and we were learning about the intricacies of the polymerase chain reaction test. And my professor probably said 33 cycles 1,000 times. We were tested on it. It was in our exams, midterms, everything. 33 cycles. When I brought out the CDC document that said that labs should run at 40 cycles, she almost kicked me out of the class. She lit me up in front of the whole class. And anyways, my experiences,
I mean,
certainly,
dude, again, like that's so arbitrary. I don't know why 33 is. It's called beyond terrain. That's what it is. Beyond terrain. I mean, again, guys, like you can do qPCR. You can do qPCR at 30, 35, totally fine. It's totally fine.
Well, influence my view in life.
Fauci himself admitted that when you run, even at 33, forget 40, even at 33 or 32, that's probably a non-viable fragment of RNA.
Yeah.
So
yeah, so I think
Idiot.
and I really appreciate you for what you do, because you're going firsthand, you're learning about this, you're looking at this stuff. I know we disagree on a few points, but I think generally we agree on quite a bit as well. We agree.
Yeah, I think it's important to focus on those, the common ground and the areas of agreement, and at least to establish a basis for discussion, right? So I appreciate your approach and inviting
what has
me
he
on,
said?
respectfully.
What is he what is the guy on the guy on the left even like contributed to the conversation? Like, what are you talking about? Like, he hasn't said anything.
With an open mind to have this discussion.
Yeah.
And, I think this is actually a great example of the type of discussion that can be had on this contentious issue. I've had a lot of bad experiences, to be honest with you, dealing with this issue with people. I've had probably more hate mail about that issue than I have on, because before I was doing stuff on like health freedom, I was, writing pretty much entirely on like foreign policy matters. And I think I had more hate mail for challenging these claims about the non-existence of viruses than I did for like writing about the Israel-Palestine conflict. So it was quite, it was quite disturbing to me, to be honest. So I appreciate this conversation and the respectfulness of it. And, I'm glad that we're able to find so many points of agreement. Yeah.
And I completely agree with that point too. With what you're saying, there is a level of hostility that I'm completely against. And I think it's just a problem in the health freedom movement. Like it's hindering progress more so than anything. We talk about how the establishment suppresses us all, whatever. Not being able to converse within the sort of health freedom community is a bigger problem, certainly indicative of a much larger issue at hand. So,
Yeah.
I'd love to do this again sometime. I'm sure we'll be able to. Maybe we can next time we can delve into some studies together. I think that could be quite interesting to do as well. Like if we get into the more nitty gritty stuff, if you're open to another conversation in the future.
Well, with the understanding that you've got more educational background in those areas than I do. I had a communications degree. I went to school for film and video is what was my emphasis. So I didn't have I had some biology and I had some. I don't think I had any. I had one class on. We discussed genetics in my anthropology class, as I recall, but I never studied any of these fields. Again, I'm not a scientist, so I don't have that background. But if you want to look at some studies, that's I'm happy to give my layman's perspective on them.
Well, I think it would give an interesting discussion, nevertheless. And to be quite honest, I think you're brilliant too. You've done a lot of work, so I really appreciate that, honestly, and I appreciate you coming on. Thank you so much for your time today.
Sure, appreciate you having me.
And I just want to hear maybe.
Oh, guys, we did it. Oh my god. Oh my. It's so frustrating, dude. It's so frustrating. I can't believe that these are the people that are running our country right now. And they're actually like advising like vaccine scheduling. They have no idea what's going on. Why did I actually try to become a real scientist? I could have just been a fake one and made 100 times the amount of money. What am I doing with my life? What am I doing? Why did I try to make a difference in the world and try to better things? Like, what was I thinking, dude? Just freaking so much easier for these guys. So much easier for these guys to just lie. It's so easy to lie. It's so easy. It's so easy for them. What? Stanley Plotkins. Sure, dude. I mean, yeah. I mean, there's just so many people, dude. That's a really good point. I really am not qualified to be a grifter. I really am. That's a really good point. Thank you. Thank you for saying that, Island. That's a really good point. I am not qualified. I am not qualified to do what they do. That is so true. So sad, dude. So sad. Whoops. No, shoutouts. Not yet. Do 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 do. Okay. Do 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 do. 1 second. I'm gonna add this real quick. I want to add this real quick. Mackenzie, thank you guys. You're awesome. I appreciate you. Thank you for backing up real science, guys. You're amazing. I also wanted to add this right here. Okay. And then we add a new tab. Sometimes you guys like fall off on here, and I want to appreciate the people that aren't here. You know what I mean? Even if they leave the chat before, I just wanted to recognize them. I appreciate you and thank you. So I was gonna do this real here. Meow, meow, meow. Do 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 do. These are the top 10 for the entire stream. Can you guys see that? Bum, ba-dum, bum, bum, bum. Yay, good. Because I want to appreciate you guys for being awesome. And I just to say thank you, just in case someone leaves, or you know what I mean? You know what I mean? Yeah, nice. Yeah, I don't know why 10th is being cut off, but I can't change it right now. I would have to make the font size. This. Let's see here. This. Boom. Way better. Yeah, that's good. Thanks, Hazel. Thank you, guys. I know. I'm sorry, guys. I am like just blown away sometimes. Like it's just, I'm so black-pilled watching some of that stuff. I have to, oh my God, it's the money. It really is the money that just kills me. It's wild how much they make. All right, guys. Well, thank you guys so much. Thank you for supporting me. Genuinely, guys. It's like an actual scientist. Like it's actually trying to do this stuff. I wouldn't be able to do this without you guys. And I just want to say thank you from the bottom of my heart for everything that you guys do, your time that you spend here going through this stuff. And thank you. Thank you so much. I feel the same way, Quangman. I feel the same way, dude. Thank you guys to all the Patreons and the super fans. Brian F, Helen, Lou. Let's see, do this real quick. One second. Thank you to the Patreons. Thank you to the superfans, Brian F, Helen, Mrs. Dr. G, Russell Bowden, Ado, Call Me Ness, Rapture, MB Human, Meg, and Dave, to the top gifters of the night. Thank you so much. Kathleen, Island Time Soap Company, thank you so much. Ducray, and to the top likers, thank you so much for 1, 10, 100, 1,000, or 17,000, like Mackenzie. Thank you so much. Thank you, Island Time, for 9,400 to 10,000 herself. Thank you so much. Thank you so much to User. Thank you for being here. Thank you to the top supporters of the channel all time, guys. Thank you so much. Aeronautical, Kathleen, Sheepadoodle, Reg Mayan, Marlis Dawn, The Miller 2024, Susie M. Vicino, Ali Arguello, and Bobby Kohler. Thank you guys so much. Thank you, thank you, thank you, thank you. You know, guys, you know what I'm going to do? You know what I'm going to do tomorrow? You know what I'm going to do? You know what I'm going to do? You know what I'm going to do? I'm going to spend so much time tomorrow working on Mava and Safe. I'm good. These people cannot win, and I will not let them win. You're going to absolutely annihilate them. They'll know our names. Peace. The work starts extra hard tomorrow. That's it. These guys are going down. All right. Love it. See ya. Bye, guys. Thank you, YouTube. Thank you so much. Thank you. I appreciate you guys so much. Yeah. All right, see ya. Peace.